I'm Doug Burren. I'm a research scientist with the USDA ARS at the Children's Nutrition Research Center in Houston, Texas, and Texas Medical Center. And I'm really excited to be here today for this workshop and tomorrow, and looking forward to this session. Just a few reminders that this workshop today is being recorded, and we will have these recordings available on the INSC website at some point. And part of the reason why I'm mentioning that is I really want everyone to contribute to the discussions today and tomorrow, because we want to capture everyone's opinions and feedback. So please don't be shy about speaking and giving your insight and opinion to these important questions that are for this. Also remember, when you speak, to speak into a microphone like this. So everyone, some of us are having, we're a little bit older than others, are having trouble hearing everything in some cases. So speak into the mic so that we can all hear it, and it does get captured also for the audience.
And identify yourself.
Yes, and identify yourself. Finally, for those who are joining online, and I hope you all are enjoying the workshop online today, to please post your questions and feedback in the chat. We are collecting those and we are monitoring those, and we would love to hear your feedback, and we will try and get your questions into the session as we go. So please do that as well. And so I think that's it. Cypress, I think that's all my housekeeping things that I need to do. But I do want to say one thing here before we start, and it's not on the script, but this meeting really began a long time ago at a meeting in Boston. Paul and Vanessa and Sharon, we were at an infant formula conference, and we discussed this concept of having something like this. And it kind of evolved, and we got Elsie involved, so Stefan was really an important part in that, and then Cypress has been a key part of organizing this, and she's been incredible in organizing everything leading up to this workshop, so I want to thank all of them. I want to thank the organizing committee for putting this together. It's been a lot of Teams meetings, a lot of thinking and going back and forth and trying to get this right so that we can get the most useful and productive information going forward to try and solve some of these pressing problems. So I am going to start off and give a bit of an overview here to session 2. And I'm going to start by talking about the Institute of Medicine report that was published in 2004. So a lot of what we're going to talk about today, and a lot of the questions that were in the SciOpinion survey that many of the panelists were queried about over the last few months, relate to this document. And the point here is, why did we create this document back in 2004? And it really stems from a request from the FDA and Health Canada to really review and identify gaps in methods currently used to assess safety of ingredients for infant formulas. And so this committee got together and deliberated over several weeks, and they created this hierarchical tree that's kind of illustrated on this figure here. If you read this document, which is a very interesting and impressive document, it's very detailed, it's very thick, very complicated. But it's very useful, I think, for our purposes today. And it really laid out the guidance, at least at that point, for how we need to think about safety. The report came out, and it was really gave us a framework to think about evaluation of formulas. And it's really 3 phases. There's this preclinical phase of looking at chemical and kind of stability issues that are common probably across all ingredients. There's a preclinical component also that involves animal studies. We're going to talk about that in detail today. And then there's this clinical part, which we talked about. We'll talk about that in the session tomorrow, of doing the clinical study that's required for most of these infant formulas and ingredient new products. And then there's the post-marketing part. So that's where the IOM started. And there's also another guidance, and I'm happy that we have a number of important European counterparts here, like Bert and others, who have, in many cases, been involved in these guidelines from the European Food Safety Authority. And they have very similar guidelines that they have created that also inform this process. There are some, I think, somewhat minor differences in terms of the criteria, the duration of the studies, things like that. But the point is that there's also another body that exists in the European Union that also evaluates formulas. But for today, much of what we're going to talk about is really informed from the IOM report. So what are the guiding principles of the Institute of Medicine document? First, we've already talked about this, and this is like a bedrock principle that is non-negotiable, and it should be obvious and intuitive, that things should be safe. And this is the starting point for GRAS evaluation, that a substance is not harmful under the intended use. Okay, this is written in law. And then also in the case of formula, it's important for us to remember that this formula represents, in most cases, the sole source of nutrition for the infant for a period of time, up to 6 months perhaps in many cases. And there's zero dietary buffer. So that's why we need to ensure that these formulas provide the essential nutrients to meet, normal growth and development. The complicating factor here from the standpoint of how we evaluate these is this last point here on the slide, and that infant biology represents a unique biological situation, and that we have in many cases an immature system, immune system, the brain, that's undergoing rapid development and growth. And so as we think about these questions of safety and perhaps suitability and function, we need to really think about these and put these in context of how we design the framework and the guideline. Lastly, and we talked about this morning, is from the standpoint of the Institute of Medicine's report, human milk is the gold standard, okay? And why is human milk the gold standard in the context of developing new formulas? Well, I think one of the unwritten or maybe well-known factors that we have to consider here is that many of us in the field of pediatric nutrition believe that in many cases infant formulas don't precisely replicate the performance of human milk. And so the question is, if we want to get formulas to be closer to human milk, then how do we get there? And so that's kind of one of the guiding principles that is unstated in many of these things that we're going to talk about. All right, so what does the IOM look like? And I've just summarized it here kind of in 4 components. I touched on these before, and it's a substance-specific tiered approach that evaluates information in some cases for sure, for these 4 areas. Number 1, we have to characterize the ingredient. We will look at its stability, its composition, its chemical identity, things like that. And then the second component of the preclinical, both of those 2 on the top of these 4 quadrants are preclinical. So the preclinical, which we're going to spend a fair amount of time talking about today, starts with standard toxicological studies that inform whether the ingredient affects organ-specific systems and whether it's safe and does not produce any harm. And then evaluate genotoxicity and so forth. And then beyond that, the IOM talks about other things like the clinical studies and clinical infant studies and post-marketing, but in this session, we're not going to talk about that. That'll come tomorrow. So No, that's not showing up very well on the slide. I don't know if you all can see it better there. So what does this tiered classification look like? So, and we're talking mainly about the preclinical aspects of this. So tier 1, as laid out in the IOM report, is essentially safety. So, and it's informed by the guidelines of the GRAS standards that are put forth by the FDA. And these, again, are things like chemical stability that I talked about, genotoxicity. But then in the case of preclinical studies, rodent studies are recommended as kind of the first place to start, either in rats or in mice. And it's a subchronic toxicity study that's a 90-day study. And for those of you who are in the industry and have been with FDA, you're very familiar with this perhaps. And then finally, also in this tier 1 category are AD and E. So the toxicologists here, absorption, distribution, metabolism, and excretion. So thanks, Paul, I got that right. So this is kind of a standard toxicological approach to evaluate, the pharmacology of a given ingredient or substance. So that's part of tier 1. So how do we get to tier 2? So tier 2, which is laid out in the IOM report, are in circumstances, and those circumstances are here now, and that's why we're here today, when you have an ingredient that is predicted to have a functional or a physiological effect in an infant. That may necessitate a tier 2 type of evaluation. And the IOM report doesn't say explicitly what you have to do, but it does describe various approaches. And we're going to talk about some of those, but they involve assessing developmental organ-specific targeted endpoints to see whether ingredients, number one, are they safe in the context of not only a rodent model. In many cases, rodent models are with animals that are not immature. They're not infant mice. They're not infant rats. And so this gets back to the context of how we need to do these studies. And that's where the pig has come in. And in the IOM report, the pig is mentioned along with non-human primates. And for the purposes of our discussion today, we're going to focus on the pig because non-human primates have kind of priced themselves out of the market. And also there's a number of ethical and animal welfare issues that have really made doing studies with non-human primates very difficult to do. So in the case of, okay, what would be an alternative or secondary animal model to consider in the context of preclinical studies, to answer this tier 2 criteria, the pig is one that has come up. And so some of the other endpoints that we're going to consider that can be done both in rodents and in pigs are doing a more detailed characterization of the molecular effects as well as physiological effects of different ingredients in these animal models, in the pig or the rodent. And then finally, we're going to touch on a few things. So new alternative models, which is a term that was invented by the FDA about a year ago, and it's become quite well known and popular. And that is models that are preclinical, that can replace the use of animals in some case, but also reflect biological function of humans. And so enteroids, human enteroids or organoids are frequently brought up in this context, but other systems and approaches like using AI and other models to try and approximate what happens in the human as opposed to an animal. So here I'm going to give you a couple slides, and I have to thank Paul for sharing these slides because I think it gives us an idea of the scope of what has been done. So this is a list of all the GRAS notifications that have been filed for infant ingredients. And you can see here over 147 notifications, over 46 different unique formula ingredients. And they are inclusive of micronutrients, macronutrients. Some of these are essential, some of these are functional. So there's quite a variety here if you think about what we need to consider in the context of these new guidelines. And then if you take some of these ingredients, and then Paul has nicely categorized these by whether they were evaluated for their equivalence to human milk. So are these present in human milk? Are they considered in that context? Were they studied and evaluated in a human clinical trial or not? And I don't know if you can see over here, but I think you can see on the screens there. Hopefully you can see virtually. And then also some of the other preclinical endpoints of genotoxicity and the like. And then finally on the far right is whether these things have been evaluated in piglets. So that gives you hopefully an overview of where we are now and how we're going to start our discussion today. So I put up this question because this is what we're going to focus on in the next hour or so, 2 hours. What non-clinical evidence is needed to establish that a novel functional ingredient is safe and suitable beyond the IOM's Tier 1 criteria? Okay, which you could put Tier 1 GRAS, kind of interchangeable to some extent in that category. Number 2, when the IOM framework triggers a Tier 2 investigation because a functional ingredient has a physiological activity, what's the appropriate preclinical model to use and what endpoints are necessary to show safety and suitability? So again, IOM didn't say these are the models you need to use. It said, here's how you need to think about it. And so what we're hoping to do, I think, with this discussion is get a little bit more of clarity on what we think as experts in this room right now should be models that should be considered, that we can say, let's use these models because we think they're more appropriate. For a functional ingredient with physiological activity is the combination of GRAS preclinical evaluation and the mandated infant growth trial. Okay, so the preclinical GRAS and the infant study, which is also part of the FDA for infant formulas, is this evidence sufficient for safety and suitability? Or does the physiological activity of a given ingredient that you propose to add to the formula necessitate a new category of evaluation that would probably trigger a Tier 2 evaluation? And we can discuss those. Okay, so that's where we're hoping to go. And at that point, I want to introduce my colleagues on the panel and thank them again for all the hard work that they put in going through the survey. So everybody they participated in the survey once, and this was blinded, and then the survey went back with the answers, and they went to a second round, and then we took a lot of those answers, we've looked at those, and a lot of that is going to inform some of the questions that we're going to talk about today. So first off, I want to introduce my colleague Sharon Donovan. Sharon, as you know, University of Illinois, has a long history in the field of infant nutrition, preclinical models with the pig, also clinical studies. Paul Hanlon, Abbott Nutrition, has a strong background in toxicology. Dennis O'Keefe from Exponent, former FDA. So we're glad to have Dennis here today. Claire Kruger. Claire, please come up from Spirits Consulting Group. And then finally, my colleague Caitlin Vonderholt from Baylor College of Medicine at the CNRC in Houston. And her background is in also preclinical models using pigs. And also Caitlin has had quite a bit of experience using enteroids. And so hopefully we can get into talking about some of those things.
Lakshmi.
Oh, I'm sorry, Lakshmi. And Lakshmi Yaruba, sorry Lakshmi, I'm sorry you're on there. Lakshmi is the research leader with the USDA ARS at the Arkansas Children's Nutrition Research Center. And her background and interests are in prenatal and postnatal factors in human milk and the role of human milk bioactives in child health and development with emphasis on the GI tract, the microbiome, and the immune system. So Laxmi, thank you for joining us, and I apologize for that oversight. Microphone. Make sure we get a keep in line here. Okay, so. All right, we're going to start off with our first thread here, and we're going to talk about safety and suitability.
Don't sit down, don't sit down, don't sit down.
Okay, there we go. All right, so what is each, can't see that, required preclinical? I'm going to go to this is our goal for where we want to go for, okay, I'm doing something different from my Casper did. So the goal for this thread is to reach agreement on two things: working definitions of what safe means in the preclinical context and whether GRAS standard plus growth trials constitute sufficient scientific evaluation. And number two, working definitions of what suitable means in the preclinical context and what non-clinical preclinical evidence, physiological function is scientifically meaningful. Yes.
Oh, sorry.
It's not going. There we go. All right, so we're going to start off here and we're going to focus on safety and suitability. And way requires GRAS, safety harm detection, and a growth tile in terms of nutritional adequacy. So for an ingredient with a documented physiological activity, whether that be immunomodulatory, microbiome related, or neurological, is there a category of safety concern that neither was designed for? Okay, and so maybe I'm going to ask you first, Claire, what would you have to say about that question? You could start us off.
It's on. Oh, okay. You can hear me, okay. So the U.S., GRAS is safety. It's only safety. There is no benefit consideration, no efficacy, no suitability. Where physiologic activities come into play when looking at safety is the thought that something that is physiologically active may have an untoward activity that is adverse. And so we're looking at physiologic activities here, not in the good way that we did this morning, but we're looking at them in the bad way that our toxicologists are going to look at them this afternoon, which is we don't want them because FDA considers them to be potentially adverse. Now, fortunately, there are suites of preclinical studies, validated preclinical studies that we have in rats, in mice, piglets, in dogs, that are designed to assess endpoints that speak to the implication that whether or not it has physiologic activity that would be adverse in the animal and extrapolate to humans. So we can actually look for those types of activities. And I have to argue a little bit with the documented physiologic activity because I think a lot of that comes from when we look at ingredients, we may be looking at in vitro studies. In vitro studies are hypothesis generating. We don't know that they actually do that in an in vivo system, much less in a human. But we suspect where we've exaggerated the exposure and controlled the conditions of the study, that we can see activities of that particular ingredient. Whether or not that's manifested in vivo, we don't know. But we then look for those. If we think that it has the potential, we look for them with the endpoints that are available in these validated models to assess whether or not we believe it's going to be something that would be later on adverse in the infant. And so I think the long answer to the short question is that it's safety, and we're going to look at physiologic activity in the context of safety. Did it do something that we didn't want it to do that had an implication later on in the infant? And we are going to use the models we have, validated models that we can utilize or extrapolate from those endpoints to potential physiologic activities later on in the infant.
Anybody else want to comment?
That was Dennis Keith. Under the statute, the safety standard is embedded in the law, so it's a reasonable certainty of no harm under the intended conditions of use. Now, there's some really important words in there that need to be defined and have some flexibility. A reasonable certainty, it's not absolute certainty, of no harm. And what is a harm? Historically, harm has been defined through a toxicological prism. And as Claire was mentioning, this idea of biological activity or some physiological activity is important in terms of making a safety determination on the exposure. The exposure is critical to making a safety decision and other conditions of use. So, for example, a number of substances can cause a hazard, but in terms of the dietary exposure, is it a hazard? And this is something the FDA deals with quite often in terms of substances added to food or in food. I do want to point out that there's another safety standard under the law for foods, and this is for foods generally. And I raise this because we're talking about here ingredients that are used in formula, but also the formula itself. For foods, the safety standard is may render injurious. Now, this is a general safety clause of the Act, but it also applies to infant formula, and it applies to foods, not food ingredients. Food ingredient standard is, as I mentioned, a reasonable certainty of no harm. So the level of evidence you get to a reasonable certainty or may render injurious is very different, and the burden of proof is very different. Whereas for the ingredients, the burden of proof rests entirely with the applicant. The may render injurious safety standard rests with the agency to. This has always been my problem. I don't speak loud enough. The burden of proof for the may render injurious is with the agency to prove that the food is unsafe. This applies to things like microbial contaminants or toxic elements, etc.
Anyone in the audience have any comments? Yes, Barbara.
So just putting together what Dennis and Claire just said, so when you look at the kind of experiments that you would do for that safety evaluation, would it pick up something that's related to the physiological activity of the substance? Is that sort of embedded in the nature of it, or does it require a different kind of testing design?
No, it picks up physiologic activities that would be adverse, because you're looking at organs and systems that are implicated in neurological development, cardiovascular, immunologic. I think the one important thing to remember here is, and I ask people all the time when they say, I'm going to do a 90-day rat tox. I say, why? How do you know the rat is a good model? How do you know that you're going to absorb it, and metabolize it, and distribute it, and excrete it the same way that your target population is going to? And how do you know you are in the right developmental period that it would be critical or very susceptible in a developing infant, so that you're testing the same developmental period in the animal model. And so I think it's very important because we default to 90-day rat tox studies, and we default to genotox batteries, and piglet models for things that are not digested. But I think that it's very important to think about, why is this model appropriate? Should I be using something else so that I can pick up what I need to pick up? And then yes, I think it does answer those questions of physiologic perturbation by when you look at everything that you can put in, and you can add some additional things that look at more specific immunologic endpoints, for example.
So maybe I'll add on to that too. So actually, I'm going to reference back to the EFSA guidance as opposed to IOM. The EFSA guidance includes a recommendation for a study for anything that is systemically available to do OECD 422 for people playing along. It is a extended 1-generation study. So it's a study in rats that's specifically designed to measure general toxicity, but also very specifically reproductive and developmental toxicity. So there are definitely models that are out there. And the other thing I want to talk about with these models is that these in vivo models, the beauty of an in vivo model where you're feeding a mammal these things is that it is both a targeted screen. Like if I could feed a rat an ingredient and look for a very specific toxicity, like I think this is going to cause liver toxicity, I can look for it. But the way that they design these studies and the way the models are set up, they're also an untargeted screen. Like you are feeding an animal something and you're measuring every single endpoint you could. Microscopic, macroscopic inspection of every single tissue, circulating biomarkers, all different blood cells that are floating around. You're looking at everything. So it would catch things that you're expecting, like I think this will cause liver toxicity, but it should also pick up those things that you may not expect, because you're not going to control what it does once it goes in that animal. If it's going to cause kidney toxicity, it's going to cause kidney toxicity. I don't have to select a model, in vitro model of kidney cells because I want to look at kidney tox. Like you get everything together in this one big model. So it's a really helpful model. In vivo, it's really hard to replace the whole thing. Yes.
So, Vanessa, if we were to take the information we just heard from our ex-FDAer and our toxicologists, we do have appropriate models in place to evaluate these ingredients. So what are we here for? Can anyone articulate? And maybe this is a question to the academics, like, what do you feel is not sufficiently being answered with the current framework? What are we trying to resolve here?
Well, I think a lot of the discussion, especially this morning, revolves around not can we add it, but should we add it? Well, why are we adding it? What is it about the functional deficiency that we believe is present because formula is not as good as human milk? What function are we trying to replicate or improve? I think those models, that is not something that these assays are designed to do. These assays are toxicology assays. But I think that as far as toxicology assays go, we have very, very powerful models. And if you look even, because FDA silos themselves, right? So the food group never talks to the drug group. But guess what the drug group does? They have to have juvenile animal studies to look at the potential in children for drug development. And so we have models, we have endpoints, we have ways to assess all of these endpoints of concern. Now, yes, if you flag something happens, you have to go on to tier 2 and something more developed or more targeted. For food ingredients, I think it would be rare if you saw something really alarming to even progress the ingredient past that, because it's alarming. So you don't really get past tier 1 very much in the food area.
Okay, I'm going to move on to the second question, because I think it kind of falls on some of these points. So functional ingredients are defined by their biological activity. So simultaneously the basis of potential suitability and potential mechanism of harm. So how should the preclinical framework distinguish the evidence for safety on one hand from evidence of suitability and activity? So Sharon, why don't you kick that one off? What are your thoughts about that?
Yeah, I guess circling back to Vanessa's question, so I think we do have good assays for safety grass, but, so I use the piglet model, and actually, I think all the animal people here, including Lexi, all use the piglet model. And so when I'm looking at this, I'm looking at things that are, we know these ingredients are toxic because they've passed all the safety studies. Where, in many ways, I talk about these as almost kind of subclinical. Like if we were feeding formulas without them, we wouldn't see any big shifts in their T lymphocyte populations, or, you know, they can't find their way around their cage, neurological deficits. But are there potential functional benefits? And that's why I think, actually the last time I was in this room, I was presenting on what are we missing if we only do NAMs? And so I showed my little piglet running around as novel object recognition and going into the MRI, and we got microbiome and immunology, GI structure and all this. So the fact is, like, we'll feed lactoferrin, and we have all these papers published. We see increases in epithelial proliferation in the gut. We see changes in immune cell populations and immunoglobulins. So I actually think by having a model, we can talk about the suitability of the pivot model, that you can test all of these different outcomes within one is actually very powerful, because we know that, as we've said before, not these single ingredients do many different things. Osteopontin's another example, HMOs are another example. So as we go to reduction, so this is where I think that we do have in vitro and in vivo models for safety, but I strongly feel that we do need to run things through an animal to look at these functional effects, and the closer we can get that animal model to a human infant, the better. So, does that answer the question?
Laxmi, I want to invite you in. You have a comment on that particular question?
Sure, Doug, thank you. I would go along the lines what Sharon said, because I also someone who used piglet models for testing different components of the milk itself. We see very much similarities, and then it is a closer model. Compared to primates, of course, primates are much closer, but as you stated earlier, it's harder to do the primate model. So piglet model is one of the most available ones that we can use, and that can be used for asking specific questions about physiological relevance, immune system, or microbiome. But there is one point to remember about microbiome is piglets' microbiome is slightly different than babies. So that outcome, when you list out all the battery of outcomes that we are looking at, some things will recapitulate human infants and some may not, but it still is the closest model.
Okay. Caitlin, looks like you've got a comment.
So I'm coming at this as a licensed veterinarian as well as a scientist, and one of the big truisms in animal modeling is that all animal models are bad, but some of them are useful. And I think depending on the question that you're asking, and what I've learned today, because I'm kind of new to this toxicology space, is that in the case of human infant formula, suitability and safety are kind of kissing cousins. And you kind of need to have both, because you're feeding one source of nutrition to an infant that is going through a lot of physiological changes. So the safety and the suitability are going to be very closely linked. And in that case, like, I'm obviously going to plug the pig model because I'm a pig person, but I think you need as mentioned, a battery of tests that do include some in vitro work to assess both the safety and the suitability. You need a mechanism of action.
Okay, Paul.
Yeah, so I agree with all those comments, and I think just to make the distinction, so what we were talking about today, and I think it's true, is when we're looking at these infant formula ingredients, we are evaluating both safety and suitability, and they are tied in that they may share some aspects of how physiologically that's happening. But I think it's also important to make a really clear, bright line distinction between how we're going to evaluate safety and the assays we're going to use to evaluate safety may or may not be the same studies that we're using to evaluate, does it have a functional beneficial effect? So and I think Claire said it too, so when I'm evaluating safety, I'm going to run my rat study, I'm going to run my genotoxicity study, and like I have very specific endpoints that I'm going to be looking at to look at adverse effects. I think
The other tie I think we have is that whenever we're doing an evaluation of any ingredient, whether that's for infants or for the general population, we have to consider all the data associated with that substance. So what is its likely, what are the reasonable mechanisms through which it's going to act? And that is really going to drive if you need to add on some additional studies on top of it. So through my years of working in infant nutrition and ingredients, we have at times selected different studies on top of that main battery to evaluate the safety of ingredients. But it's really driven by what is that ingredient. So I was responsible for or involved in submission of additives to Codex. So JECFA evaluates the safety of additives. The two additives I'll talk about, and sorry if you called into the roundtable, you hear it again, but one of them was xanthan gum. It's a super long polymer. It's not absorbed. It goes right through the gastrointestinal tract. So just like I said at the beginning, every ingredient could have a physiological effect. So what is the reasonable physiological effect of xanthan gum? It's going to be on the gastrointestinal tract. And in that case, we selected a pig model to look at the safety of that ingredient, because that is like, look through what are the endpoints. Let's select the correct endpoint to look at that very specific function. The other ingredient we worked on was ascorbyl palmitate. So ascorbyl palmitate, it breaks down in the gastrointestinal tract into ascorbic acid and palmitic acid, normal nutrients that are, your body knows exactly what to do with them. In that case, we used, you could call it a NAM, like they in vitro looked at whether or not it was digested by the enzymes that are typically found in the digestive tract of infants. And they found complete dissociation, and based on that, they could say there's not going to be any concern with this past ascorbic acid and palmitic acid. So it is always critical to think about what is that physiological potential that these ingredients would have. And I think we do, I don't know, I think we do as a toxicology field do a good job of like incorporating that into our systems. But then it's really leveraging those validated models when we have them, things like the OECD 408 and 422, getting those endpoints. Again, they're non-targeted screen, trying to find anything else that may possibly happen. But on the safety side, you can build on that. I think we've got some other good work that it's also been referenced here today. Are there other endpoints that you want to build in? If you have other potential reasonable effects like immune effects, do you add in some endpoints of the clinical? There's other things you can do, but it's really going to be driven by your comprehensive risk assessment and your review of the literature of what different organ systems could be impacted.
Okay, so I think we're going to move on to our second thread, but before we do that, oh, Bert, sorry, Bert.
Thank you, Bert Koletzko, LMU Munich. I have a question to the panel in relation to the Q1 that you showed, where it states you need grass and you need a growth trial. And we are all aware of this. It's like the dogma from God. With every substance we evaluate, we need a growth trial, right? But there's a lot of components where we would say we would not expect any informative information from a growth trial. Obviously, growth trials historically were very important when we modified protein, when we had other things that changed growth. But if we had a substance that influences immune response or neurodevelopment, where we have absolutely no reason to believe it could interfere with growth, and if we have an animal study that shows that even at supernormal levels that we would ever give to an infant, there's no effect on growth, can we consider that a growth trial would not be necessary? You may say, well, you need a human trial anyway, you need a clinical trial to evaluate effects in babies, but it's not necessarily the same as a growth trial where you have to exclusively formula feed from early on, whereas in the real life, we often give formula on top of breastfeeding or after breastfeeding at a later age, and we would like to evaluate an effect of a component, not an exclusively breastfed, formula-fed baby from early on. So it's a real question that also raises maybe ethical issues. Can we really justify exposing babies to such trials when we don't have a hypothesis for this? So my question to the panel is, what is your view if you have that situation? You have no reason to believe there's an effect of growth. There's no effect on growth in a preclinical trial. What do you do?
question. It's a really good question, and it's something I've been thinking about. So one thing I think we need to be clear about, I'll get closer to the mic, so you can hear me. The safety assessment on the ingredients is primarily based on tox studies, right? The studies around nutritional adequacy are done on the formula. So that safety assessment in terms of the ingredients has been done. The nutritional assessment is part of the formula assessment and whether it's nutritionally adequate in its entirety. And it's very important to be clear about that the nutritional assessment is not part of the ingredient safety assessment. Does that help?
But to your point, Bert, if there was no reason to think that an ingredient would have an impact on nutritional adequacy, I can tell you what the regulation demands, which is the regulation demands you still have to run it. But scientifically, I think we could have a discussion about whether it makes sense or not.
Well, I would ask the question, is it possible that if you show that an ingredient is safe in a preclinical model or two, and you don't predict that it's going to affect growth, is it possible then that a human clinical trial is useful just as a last final screen of safety? That's probably not the way to think about things, but I don't know.
Well, I mean, I think that's correct in a way, in that, you know, as you said, we're looking at the formula and not just the ingredient. And, you know, just from the perspective of a toxicologist, I think the most sensitive endpoint of an impact on health is loss in body weight. That is, in animals sick, they lose weight, right? So you may not know what got impacted, but they're losing weight, and that's a really big flag. So the fact that you do a growth study and you have those infants eating that formula, you're demonstrating the nutritional adequacy and the safety of it by showing that they're growing appropriately during that time frame. So I think it's a good last in your target animal, right? You're human. You are doing that study to show that within the context of that matrix in the formula, still nothing went awry.
Okay, I think we're going to move on to our next thread, if we can. Hold that thought, though, Dennis. I mean, I want to kind of keep us moving because we've got 5 threads to get through, a few more than our first session. So our next thread, well, we've got our test, our Slido question. So is there a category of safety concern specific to a functional ingredient's intended physiologic activity that the existing GRAS preclinical package was not designed to detect? So go ahead and enter in your responses there. There's 4 different responses. And Dennis, why don't you were going to mention something. Why don't you go make your comment here while we're watching the votes come in?
Thanks. I was just going to mention, you know, and Barbara probably knows this better than I in terms of the infant formula regulations, but you know that the authority over infant formulas for FDA was driven by an adverse outcome where the formula was not nutritionally complete, and I think there was a chlorine or chloride deficiency in the formula, and that's what drove the legal authority for the FDA to have authority over the formulation itself. And in fact, they're supposed to be looking at new production facilities as well when a new formula is being made, and going through a pre-market review of the formulation, the new formulation on the new production line. So that's sort of the mindset or the framework in terms of that requirement for the clinical trials.
I would add, I think the issue that's being raised here, I would put in the parking lot because it would require a change in regulation to implement. So it's not that it's a bad idea, but our deciding it is not going to have a huge impact here. Anyway, that's sort of my feeling. I do, in listening to the detail of these studies that are provided, I know that the tox, you know, it goes to OFAS. That's where it gets handled. I find myself wondering how much information goes to the infant formula staff so they understand the full scope of what has been done when they're starting to look at that notification. Anybody can answer.
I mean, as you know, it depends a lot on the integration of the information sharing between the two groups. And, since we are at FDA, the foods program has been reorganized, and I'm not sure quite what the communications lines are currently.
Yeah, and some of it might be to the industry. How much do you make sure the infant formula group is aware of? I did want to also point out on the growth monitoring, we do now have a National Academy study that's looking at those quality factors. Will it change anything? Who knows? But, there's usually a long timeline in that as well.
Okay. On that, we'll move on to our next thread. There we go. All right. Thread number 2, the mandatory preclinical package that is what is required, what it should be, the preclinical. So our thread here, our agreement would be to the provisional mandatory preclinical safety package with explicit scientific justification for each element, including acknowledgement of what each study does and does not establish relative to GRAS. And then hopefully the specific scientific criteria that elevate the neonatal pig study from a substance-specific option to a substance-specific required, whether we want to get to that point. So our first question then for thread 2 is, what non-clinical study should every novel function ingredient be required to complete before the mandated clinical growth trial? And I'm going to put this first question up here. And I would just make a couple comments from our survey that this was one of the questions where we had, on one hand, almost universal agreement that the 90-day rodent study was probably a good requirement, but where we had disagreement among the panelists was to what extent should a piglet study be required and under what conditions should a piglet study be used. So question 1, the 90-day subchronic rat study commands near universal endorsement, but uses adult rodents at supraphysiological doses and was not designed for neonatal specific physiological systems. So for an ingredient with a physiological activity in the gut, the immune system, or neurological, what can the 90-day rat study establish about safety of that activity? And what does it leave unaddressed? So I'm going to pass that one over to you, Caitlin. I'm going to let you and Laxmi kind of comment on these two first, on this first one.
So I am not a mouse or rodent person, so perhaps I'm going to give you a biased perspective. And this also feels like we're advertising the pig model with 3 pig people on here. I promise that's not how this was planned. It just happened. So one of the things that we think about in animal modeling is making sure that the model that you're using appropriately reflects the physiology of your target population. If the model that you're using does not reflect some of the unique facets of your target population, it's useless. You have figured out how toxic a ingredient is in a mouse. So you can take that to the mouse food corporations and make a lot of money, I guess, in some countries. What I think needs to be important in this context specifically is that if you're going to feed something to a neonate, you need to test it in a neonate.
All right, so I'll weigh in on this. So I think there's another element here, which is that we have some of these studies. So I keep saying OECD. So OECD is this international body that comes together and they validate studies. So they say, if you run this model using these very specific parameters, you can trust the results of that. You'll have a clear binary, things are looking good, they're safe, or they're not looking safe. So the OECD 422, which is the extended 1-gen study that does include neonatal animals, the one that is recommended by EFSA, even though it is rats, like they are saying, if you run this in rats, and that's regardless of it's used in food industry, in the drug industry, in pesticides, et cetera, they're saying that these results are translatable to the human model. So they've done that validation of this is the appropriate model to do it in. So I definitely think that everything you said is true. Like you always want to select the model that best represents it. And I think the good news is like, that's what the OECD process will give you confidence in, is like, you are selecting the correct model to do it. So coming maybe quickly back to the question that's on the screen. So where do I think every single ingredient needs a 90-day rat study? I think if we look back at the color-coded slide of which ingredients had it, the answer is not all of them. So I agree that not all the time do you need this study. And it's a very unsatisfying answer of it always depends and everything should be case by case. But it always depends, and it's always case by case, because some of the ingredients that you see on the GRAS notification list, I think we would all look at those and say, like, why would I run this in rats? Like, it's whole milk powder. Like, who's okay, I see dairy people in the audience, but like, who's going to really run whole milk powder through a rat? I can tell you right now, rats live on garbage. They will live on whole milk powder. So I think every time you do it, you do want to do this case by case. You want to look through what are the reasonable effects that are going to happen, and you want to select the correct model to take a look at those effects.
Okay, Laxmi, jump in here. What are your thoughts
So
on this?
I'll add to both of them. A rat model is, I mean, initially it's good to know the toxicity aspects of the ingredients that are being tested, but beyond that, it doesn't really recapitulate a lot of things that an infant has. So again, like Caitlin said, I didn't really plan it being piglet being advocated, but that's what I think is the closest model to the infants. So that's how I think about it. And in terms of physiological responses, I'm not sure if they really recapitulate some of the things that we look at, or the GI tract or immune system or any of the brain studies. So that's how I would think about the rat model as an initial test.
So I have to stick
okay.
up for rats. I'm sorry. I find it's a weird position to be in, but you know, there are, and again, I agree with all these comments, because you have to find the model that is appropriate for the human, the developmental stage and the metabolic handling. And so it's not always necessarily going to be the rat. However, we do know from extensive research over decades, and FDA and the drug group and food group have used these models, that because we understand what the critical period of development is for organs and systems in different species. Now, the rat is harder to work with as a neonate, but pre-weaning, than a piglet, because it's little. So the practicality of it, and yep, I've done little neonatal rat. It's hard, but just because it's difficult doesn't mean it can't be done, and doesn't mean it's not appropriate, because you can give them exaggerated amounts during appropriate levels, and you can demonstrate that their metabolic handling of the ingredient on a case-by-case basis is correct. And when you get that, it's important to remember that we apply safety factors to those results. So we're not saying we fed the rat X amount, that's how much a human can get. We apply safety factors to go from chronic to, subchronic to chronic, to go from rodent to human, to go from, non-sensitive to sensitive human. So there's a lot of safety margin built into those calculations from those rodent studies.
So I'm going to go back to the pig. So I think there's a couple concepts, and I'm not against doing toxicology studies in rodents, mainly for the reason you're talking about. The FDA knows what to expect. They know how to interpret that result. So over the last 2 years, FDA has reached out personally and said, we don't have people who can help us interpret pig studies. And they were getting GRAS applications, and they're like, well, it doesn't look like this pig is growing correctly, or how do I interpret this? So one of the things we've been doing is, so with Ryan Dilger, we now publish piglet growth curves. So just like you would have an infant growth curve, we have them for artificially reared piglets plus sow-reared piglets with different fed ad libitum, fed per kilogram body weight, some challenges like choline deficiency, iron deficiency, you see they fall off. So what we're trying to do is help to build those standardized models, so the FDA has some standards to report against. Where Ryan's doing the same thing with clinical values. He has an MRI brain axis, just like we have for Atlas for humans. So I think the biggest barrier right now is that there aren't staff at FDA who understand the piglet model and how to interpret it. And it's uncomfortable because it's not like you have this cookbook and everybody does it, we know the results. But I would say is that we have done many a study where we incorporate a safety study and an efficacy study at the same time. So we work with a board certified veterinary pathologist. We get all the lab values, we send them to our vet hospital. So we can build a safety study, but also be doing MRI or doing novel object recognition or looking at the gut microbiome. So I think it might be a little early for the pig to become a safety toxicology model, but it's certainly there. They're already used for toxicology straight out. But I think the biggest barrier right now is having people at FDA and having standards that they can agree upon. But I would basically, if somebody's doing like a plant-based formula and the pigs are like starting 50th percentile and then suddenly they're dropping off, you would be able to see pretty quickly. And unfortunately, so actually 2 years, maybe 3 years ago now, we did, prior to a swine and biomedical research conference, we did a one-day workshop on using the pig as a model to screen bioactives. And we tried to round up the troops of people who worked in this space to, again, think about how do we bring more scientific consistency and practical consistency to how studies are run, which I think is probably the biggest barrier right now.
I just want to just add to what Sharon's comments. What Sharon is talking about is really important because on all of these slides, we're hearing about GRAS and we've talked about the safety standard, but GRAS stands for generally recognized as safe. And there's two components to meeting that criteria. One is that the information is publicly available and that qualified experts would agree on the safety assessment. And not 100%, but it would be a consensus. So if you don't meet those two standards and you think you meet the safety standards, you fall into the food additive space. So Sharon's work is really, really important to get it out in the public domain. Thank you.
Anybody out there got a comment? So I think just maybe to add to this, is there a possibility that to do a safety study in a rodent that you might miss something that if you did it in a pig, you might see from a safety standpoint, because it's a neonatal animal model that's maybe more fragile from an immunological standpoint or something like that. Is that a possibility?
I mean, I think it's always a possibility, but again, if you look at the paradigms that have been set up in the regulatory frameworks for other places like drug and petrochemical, agrochemical, they don't recommend other studies. Like they say, run a neonatal study in rodents, and then that's sufficient. Again, I think it does come back to case by case. So are there some studies where you want to look at the pigs because it's a better model for gastrointestinal or some other model? I think you can certainly find cases where that would be the case, but I think you can identify which cases those would be based on the characteristics and the composition of the ingredient you're talking about.
Yeah. Okay. Dave.
Yeah, I related to what Doug was saying. I mean, if you think about like necrotizing enterocolitis for like preterm studies, then things that are already GRAS status have been associated with necrotizing enterocolitis, at least in piglet studies, right? So does that mean that a higher threshold of safety would be required? Because obviously that safety concern wouldn't have been picked up in the 90-day rat toxicology study, right?
I'm not quite sure I understand the example you're talking about.
Yeah, so some studies using like preterm piglets, if you like, sometimes what they do is they'll like feed a infant formula.
Okay.
Versus a human milk, and the infant formula will increase the the frequency of necrotizing enterocolitis development. That's like a Paris Angild's lab will use that model quite a bit.
So I'm going to
yeah,
jump in
yeah.
because I use the preterm pig model mostly, and I study a lot of necrotizing enterocolitis.
Yeah.
If you have an ingredient that you are absolutely certain is safe, and I can feed it to a preterm pig, and it will very, very sensitively tell me what's wrong with that ingredient. Because preterm pigs are extremely delicate. They have an immature immune system that's also hyperinflammatory. Their gut doesn't work, their brain doesn't work, their lungs don't work, but they are a good way to test something if you want a very, very sensitive readout for safety in a very specific model, because they're almost like a different species.
Yeah, and I think that's why we actually initially said we're going to address only term, because preterm is you're looking at how abnormal compared to the abnormal. Preterms is a whole different situation.
They're weird.
They're weird. Term infants it's a very different situation. But preterm I can see, that may be, a situation where the utility of that kind of model is exquisitely sensitive and more appropriate. But preterms, that's much more difficult question.
Can I just add real quick, Vanessa?
Yes, Vanessa.
So again, going back to the same sort of question I asked in the first thread, where it doesn't seem to be a problem, like it doesn't seem like we have a problem with our models, with our safety evaluation, and yet we're still here because obviously there is a problem, or at least there's a problem on the perception of how our safety studies are done or what they can accurately tell us for these at least functional ingredients. And this is a question sort of side by side with the academics and the toxicologists. Toxicologists are saying toxicology can answer all safety questions, period, for ingredients with known physiological activity. Do our academics agree? Like, are there instances, or is there a different definition of safety with these ingredients that have known physiological activity? Lactoferrin, lysozyme, we haven't talked about that one today, right? Why is there still inability for general community to accept safety of these ingredients that have demonstrated absence of toxicological effects? And yet there's still, what is the outstanding question? If there's demonstrated absence of toxicology, what is the safety concern still at hand that toxicology is not able to sufficiently answer? This is open to everyone, but.
You know what I'm going to do? I'm going to go to the next question because it's related to your question right there. Okay, so the question here is one of the experts, and I'm not going to let you guess about that, argued the needle pig should be mandatory but not substance specific on the grounds it's the best current available model for physiological systems. So majority favored a substance specific designated. So what specific scientific criteria should trigger a neonatal pig study to address issues of physiological function and not necessarily safety, perhaps? Is that getting to what? Yeah, okay.
So maybe I'll go back to your roadmap question from the first session. So like, what is the roadmap to like really defining what do we need going forward? And yeah, I will speak for the toxicologists that I think we've got really nice guidance that was established by IOM that I think probably needs a once over. I think we have an updated guidance that came out from EFSA in 2017 that I think builds on the new information that happened in that decade and a half. When I look through the GRAS notifications that I've gone through and all the safety data that was submitted as part of that, I think it really does start to spell out a picture of what are the types of data that you really would need to establish reasonable certainty of no harm for these ingredients under their intended conditions of use. I think it would be, there are certainly some scenarios where you may need something past just a extended one generation rat study as well as the gene tox. Some ingredients may not even need that, but I think if you could get together a group of experts to really look through what are the specific endpoints we're concerned about, do these studies address those, what are the special situations where we would want to go beyond that, and like let's map out, here are the specific studies that could be run either preclinically or endpoints that could be added to a clinical study, and like really let's say evolve the IOM guidance, the EFSA guidance to get to a point where you I'm not going to say checkbox because it won't be checkbox. It's still going to be case by case, but like really get it to more of a framework where people could work within this framework.
I was gonna say, I think you asked the question, why, if there's, we've heard that there's universal agreement that these studies are good, why are there still questions? And I think one of the issues that we deal with is kind of a change in perception at FDA from studies or submissions that we used to do 15 years ago, where, there was no mention of function or activity or that was just something that's not, it's a safety. Just get rid of that. Just eliminate that table. And so everything was looked at under the lens of kind of structural parameters and not functional consequence that related to the findings for those parameters. Now there's a recognition that a lot of what they're looking at has function. And so I think we have to change our explanation of and education of what the implication for those parameters means from a functional perspective in the animals. And that's why when we look at these kinds of studies, which we do all the time, we look at the studies and we go, okay, this parameter was changed. What does that really mean? What else did we look at in these animals that told me whether or not that had an implication for an adverse physiologic consequence? And so I think some more explanation and interpretation and context around that would help people understand the value and the ability to extrapolate from those to humans.
Dennis.
I would totally agree with that, and it's really important for the sponsor to provide that information to the agency, because what the agency is doing in response to a GRAS submission or a food additive petition, it's creating an administrative record that provides a scientific and legal basis for any action. And if that administrative record is not complete, the agency is open to legal challenges in the courts. So it's really important for the sponsors to help the agency create that administrative record, and you can really protect yourselves.
Yes, Barbara.
I really appreciate this discussion because indeed it comes to me, how do the tox studies address the physiological activity and who needs to see that information? I think from a safety point of view, we assumed it was OFAS, that they need to see that information, but it may be the infant formula team also needs to see that information, or at least understand how those particular tests address the physiological activity. And again, I want to point to the tension that exists. These things are added so the product can have a claim. There will be a claim on the product. The European Union, EFSA, they review structure function claims. They review the kind of claims and decide whether to allow a claim or not. We don't. We basically say, it's your responsibility to have the substantiation for that claim. We can go after you, but it's your responsibility to have that substantiation. So it's not surprising to me that the infant formula team remains concerned that what does this product mean when it goes out with that claim on it?
Yeah, I think one of the things that I'm trying to clarify here is, so what justifies going to a tier 2 type of evaluation? And we're talking about ingredients here. We're not talking about amino acids and essential nutrients. We're talking about functional nutrients. And so what about that functional nutrient? It has a physiological role. It has a function. Is that sufficient to then say we need to go to tier 2? And then on top of that, do we have to have a model that is capable of identifying the effect of that function or its physiological action? And I think we're all probably saying, oh yeah, do it if you think it's appropriate. But what maybe we're trying to identify here is, should we make this something that is necessary or we're recommending to do as a guideline? Does that make sense?
I think not if we think it's appropriate though. It's like if the data demonstrates it's needed.
Right.
If that, maybe I'm slicing the bologna thin, but like that, it's not like I'm choosing to or not based on my personal opinion. It's like the data is driving me towards this is an endpoint that needs to be explored because of what we know about
Right.
the compound itself.
So let's take maybe one of our favorite proteins that comes up a lot here, lactoferrin, right? So lactoferrin, we know it has a function. Would it perhaps be appropriate and suitable to test the effect of lactoferrin in a pig model as opposed to a rat model, because you might be able to assess its function more appropriately?
Or is it the piglet model that's right? Or is that, so if you look at the Callahan paper would suggest that you would incorporate endpoints into a clinical trial to get at those same endpoints. Right. So the rat model does look at immune effects. So like It will look at effects on immune tissues, but I think that is the good discussion of, okay, based on what we know, if it's an immunologically active substance, what are the other endpoints that you may want, that not you may want to, but like that it forms that you should be running on that ingredient. Mm-hmm. Sorry, I'm stealing your thunder, Dave. I see your hand up.
Yeah, I was just thinking of a example that might be relevant for this question, which was, I think that I remember reading on one of the like FDA responses for one of the recombinant proteins that was being put forward, that there was some concern about the potential for like autoimmune development, right? And that was specific to the fact that the protein sequence was like identical to a human sequence. And so if that's the case, then you couldn't use necessarily like a human protein sequence to assess potential for autoimmune development in a rat, because it wouldn't be matching to their particular sequences. And in that case, like potentially like a tier 2 study would have to be a human study to assess that, right? Like even beyond a piglet study, because that alone wouldn't be sufficient either.
Hey everyone, this is Ross, Ross from Helena. So yeah, so for human lactoferrin, I think that's the case in point is, these tier 1 for the longest time, the feedback is general tox, is not going to pick up some of these immunological signals or toxicology that they were concerned about. So nevertheless, I guess Helena, I'll put a plug in. They did do these tier 2 tox, which is part of the rodent model, which I think are validated measures. They are quite validated, and the TDAR, the T cell dependent antibody response, and then immunophenotyping, kind of 2 separate ways of looking at how the immune system from a cell type, balance, and then also functionality. That was showing that, hey, there's no immunotoxicity happening, but to the call out from FDA, hey, if you have an immune, a protein that is human, the only way to determine if there's truly an autoimmune risk happening is to look at it in the species that it's native to, and that's the human. And because, of course, human lactoferrin we have in our saliva and it's all in our bodies. So could you break tolerance to that by giving exogenous? And so in that case, I think there absolutely is a reason to run a human study to ensure that there's not going to be this breaking intolerance. And the good news for human lactoferrin, at least from Helena, is that the adult clinical studies showed no increase in antibody titers against the human form of lactoferrin. So I think in certain cases, and that gets back to the case by case, right? Every ingredient, that kind of puts the burden on the company that knows the ingredient best to be able to define, okay, hey, what does the literature say? And then rally some experts, right? And get the data published, particularly for grass, to start building that wall of general consensus and general recognition.
I think it's actually an interesting case of, and I think the studies were well done in terms of being able to address that question, but it's a question that came about from a theoretical precautionary principle approach, in that there's a reason we don't give proteins drugs by the oral route, because you break them down. And there's zero cases in the literature anywhere of an orally administered protein causing autoimmunity. So this is a philosophical, theoretical question. So that gets to, at what point does a question beg have you established reasonable certainty? And at what point do theoretical questions kind of factor into your approach? Because to me, that was just a very interesting example of why would this happen? It has never happened. It's never been reported to happen. Nobody can come up with an example. How would this even work? There's no mechanism by which you can even suggest this would happen.
So I just have a quick comment. I always remember the words of Hugh Tucker, a lawyer at Mead Johnson many years ago, who said, "You never conduct a clinical trial until you know what to expect. You want no surprises." So this example, if you are going to move your immune outcomes to your clinical trial, you'll get blood. And what we can do in the piglet, we can give mesenteric lymph nodes, Peyer's patches, spleen, thymus, blood, so that you can actually be clear on, well, maybe there's something in the intestinal immune, mucosal immune system, but there's nothing in the thymus or the spleen. So I do think it's that extra measure of the ability to have samples that you could not get in a clinical trial. And if you really think, again, case by case, that there might be an immunological response, it's that extra safety level in a way.
Okay, I think we're going to move on to our quiz question here. All right. Should the neonatal pig study be mandatory for all novel functional infant formula ingredients or substance specific? Cast your votes.
Either way, you're going to make half the panel cry.
Rats versus the pigs. Okay.
Is there anyone in the audience that might have a comment while we're watching the votes come in here? Yes, Dave.
So I think there's also the consideration that we have to take into account. Like, as you were saying, the pig model is a model, and we don't necessarily know that it will capture all of the things that we're searching for. So it's possible that the pig model alone is not sufficient, right? Like, you may still need some sort of human safety testing in addition to a growth monitoring study. In I study protein digestion. What?
Session 3.
I know, yeah. But I study protein digestion, and nobody has actually validated that protein digestion in a term infant is the same as in a term piglet, right? So like, just that very tiny specific question hasn't been answered. So
the pig and human immune systems are about 90% homologous. The pig and the rodents 10%. And even NIH says we should not be using rodents as a model for the immune system. So yes, nothing's 100%, right, Caitlin? But I think if you want a good immune, this is why they use pigs for organ transplants to humans, right? So it's good. It's good as we're going to get in the model.
Yeah, we're only at 3%, Sharon. That didn't help.
Well, no, I don't believe it's mandatory.
No, I'm kidding.
I can change my answer. So this is where I think, and I know NAMs are a hot topic in a lot of places, but one of the benefits of using in vitro models, the novel animal, non-animal, whatever you want to call them, I think is that you can use human-derived tissue, or in some cases infant-derived tissue, for specific mechanistic questions. And I think that's where you can do like I don't want to say a tiered approach because we'll get back into the IOM, but I think if you can ask questions in a petri dish or an organ on a chip or even in a zebrafish that you can later take on to other species that may better recapitulate what you're looking for. That's where you have to kind of dig into what's available now, because there's a lot of things that we can do, not just for hypothesis generation, but also for function, safety testing, et cetera.
Yes, we have a question in the back.
So has there been a case or a situation where these models failed, has there been a situation where an ingredient appeared safe using the rat model, but then the pig model caught a problem?
Not that I'm aware of.
I was thinking about you mentioned xanthan gum, like remember the Simply Thick situation where babies were getting neck from the thickening agent, and then it was declared that now it's not safe for infants. Like, had that gone through these processes and it failed?
I think that was a micro thing though. I think that was a contamination thing. Trying to look for help from the audience, but I'm not aware of. So I reviewed those 146 notices to the FDA database, and as far as I'm aware, none of those ingredients has gone backwards. Like, based on the data sets that they had to support their safety, nothing has come out suggesting that there's a safety concern with those ingredients. So I think it is another endorsement of the models that we do have, that they are robust models. I think the other thing to remember is that when we're talking about food ingredients, for the most part, we're talking about things that, even though they do have some physiological effect, they're much less physiological potent than some of the other chemical spaces that these models are used for. So if you're talking about drugs or petrochemicals or pesticides, when you're administering those in animal models, you're dosing until animals start dying. And I have never had studies where there was a test article that caused death of animals. What we're dealing with just does not have the same physiological activity as some of these other things. So we're already starting from a baseline where these things are pretty safe. They're nutrients for the most part. So we're in a very different chemical space. So thalidomide, probably people have heard the story about thalidomide. Like, yeah, there are cases where drugs have gotten approved that later had to get withdrawn, but those are highly pharmacologically active substances as opposed to the chemical space that we're talking about in food ingredients.
Okay, yes, one more comment.
Hi, Leanne Redmond. I was just curious, Sharon and others there, are there some specific criteria already developed around what these piglet studies should look like? Litter size, the condition of the animal, like at the beginning of the study duration, these sorts of things?
No. And I think that's the problem and why we did that pre-conference, because every university has built their own infrastructure for pig models, and we feed them differently. We may not feed them the same, like we use a sow milk replacer formula that we kind of humanize a little bit. But see, that's what I was bringing up before. And I think one of the questions we have coming up is, should there be standardization? And we're doing part of it, but we would really love to get the whole pig community internationally to come to some standards. And I mean, the FDA, we sometimes feel like they're holding us to a different standard because they asked, well, these people have to drive their piglets 4 hours. We get ours, from a farm that's 5 minutes. But like, nobody asks if your mice come from Jackson Labs and they get shipped to San Diego versus, New York. And so I think that there's a lot more leeway because it's accepted and it's known in rodent models. And also rodents are extremely inbred. And I think that's right. If you're going to see something that doesn't line up, it's because, even our pigs are pretty, they're still outbred, but they've been genetically selected. Yeah. And it matters. And I'll give one more. So I have a former colleague, Kelly Tappinen, who did a short bowel piglet model. Another advantage of pigs, you can do these cool models. And the animal scientists on the farm decided they wanted to change the genetics to go to leanness. And she had to kind of redo her model because they were selected genetically for rapid growth and leanness, but it affected her gut resection model. So we know if you want to very easily, I think, accepted and reproducible, the rodents are your game, but humans aren't like that, right? So to have some variability, I think, actually provides another level of insight into what would be your standard deviations?
Yeah, I just think it's an important comment to include in the paper at the end of the day, right? If the group has sort of come to consensus that we think that this is a good viable model moving forward and one that can replace the non-human primate, I think we also need to just identify that there's still some gaps here.
Well, and everyone loves the primate. And when I was at Davis, I worked with the primates, but I don't think they're necessarily the best model. And most primate mamas have one baby. Our piglets are actually, we have typically 12. We've had some that, because they've been bred for credibility, 18 piglets. So we from the same sow can, randomize them just like you would do with rodents into all the dietary treatment groups. And you can't do that with, with monkeys. And so, yeah, monkeys I think have a purpose, but I think we can do piglets instead of monkeys.
It's a cost and feasibility issue because pigs are expensive as it is. So, and that it's like the pig is somewhere in between the rodent model and this issue of, well, pigs are not, Sprague Dolly rats. They're not all inbred. Well, you could say, well, that's an advantage, even though it might create some variation in your outcomes, because one litter from another litter, even though they're from the same genetic background in the mothers. So I think, there's considerations on both sides of this, but I think the point that's going to come up, and we're going to take a break right now after this, is that we're going to talk about references. And I think that's an important thing to consider with regard to the pig. So with that, let's take a break. And why don't we come back at 3:30? We're going to take a little bit of a shorter break and come back at 3:30. Go ahead and get started with thread number 3, and that is entitled Model to Outcome Matching. So which model for which outcome? And our consensus is at least 2 to 3 provisional model domain endpoint pairings, the appropriate model for a triggered neurocognitive tier 2 evaluation, or for a microbiome suitable evaluation. So can we get to consensus on how we want to identify a model to match an outcome? And then what IOM framework currently lacks and what modernized version could be specified? Okay, so here's our question. Question number 1. So the IOM framework specifies that tier 1 signal can trigger tier 2 in the case of neurocognitive evaluation, but provides no guidance on which model is appropriate. We've kind of been talking around that. So is the neonatal rat behavioral battery scientifically adequate, or does the neonatal pig provide a meaningful, more translatable data to that question? And if the piglet returns a dose-dependent neurological signal, is that finding interpretable without reference data? So that's a mouthful. So I'm going to start off with that, and I want to bring Lakshmi into this because she's done some neurodevelopmental studies in pigs and rodents. So Lakshmi, why don't you start us off?
So again, I guess I sound like a favorite for piglet model. Sorry. I'll do my best here. So the way I view this is from the experience, from the literature, from what I've heard from everyone here today, and then all the literature as well. Neonatal rat model, yes, it's a good first step for toxicity and toxicological assessments. And then in terms of translatability, when it comes to specifically microbiome is not necessarily very similar in piglets versus infants, but it does provide a good translatable data. For example, we have fed pigs with human milk versus formula diet, and when we compared some of the specific outcomes of the GI tract or some of the other like growth data or any of it, you could compare, and we do see some of the same things in infant clinical studies, that are fed breastfeeding or formula, and immune response outcomes or any other growth outcomes. So there is a translatability from the piglet model much closer to the infants. And when it comes to behavior stuff, Sharon has done more on the piglet behavioral stuff than any of us here. Infants translatability, I think there is, from the literature and from her papers, there is cognitive assessments that were translatable So that's also a good sign for piglet model. And microbiome, there's differences. I'm not saying they're bad or they're good. There are differences with the piglet model versus infants that either receive feeding or formula diet. Specifically, bifidobacterium is very predominant in infants. When you look at the literature, including our studies and everything that's been published in piglets, you cannot see a bifidobacterium as much abundance as you would see in an infant in the breastfed or formula feeding. So I think there's some things that we have to admit. There's limitations with the models, but they do provide things so that we can test for safety, suitability, and then in terms of physiological systems so that are readily available, that we can address some specific research questions and mechanisms. So that's how I think about the appropriate model.
Neurodevelopment, Sharon, brain behavior. How well can we use the piglet or the rodent to match neurodevelopmental endpoints?
So all the neurodevelopmental work at Illinois has been done by Ryan Dilger in animal sciences, but we typically will conduct one study and he'll do behavior and MRI scanning. I'll do immunology, gut, but it makes we can work along the gut-brain axis. So one of the things I mentioned with growth curves and things, we've worked very hard with the piglet model because of the size. We can use a standard MRI. And so he's done scans from like 2 days of age till they won't fit in the magnet anymore. As I mentioned, developed the atlas. So it's something that's freely available. So if other people are doing this. So the idea is like, what can we do that is translatable to the human infant? So when he started the neurocognitive, I mean, we looked at the rodent literature. He's tried 8-arm radial maze, T-maze, eye blink conditioning, most recently novel object recognition, because we understand the mechanisms, and you can do that in human infants. So I'd have to say that the MRI is very sensitive, and that's where we've done a lot of studies looking at HMOs and prebiotics, symbiotics, and you can see differences in regions of the brain. And again, the nice thing with MRIs, you can do repeated measures, so you can look at trajectories. So the novel object recognition is a little bit trickier, and one of the advantages in some ways, but disadvantages, piglets are really, really smart. And so we see great effects in deficiency models like choline or iron deficiency. When you knock them down and then provide adequate diets, you can see compensation. But again, novel object is like a 50-50. So you're just looking at, are they spending more than 50% of their time with the new object versus the object they're familiar with? And so you have to have like a larger number of animals. You have to power to see that. And you would also, again, that would be something if perhaps a rodent model, even an adult rodent, for example, like with a human milk oligosaccharide, suggested that there might be some learning and behavior. You can do some of those in the pig. So I think, yes, I mean, it's certainly from MRI and also from behavior, it's there. But again, it kind of gets into the monkey stuff where not everybody has a facility when they can take a pig up to the MRI, and do all that. So it does kind of limit, where you can go to do these types of studies. Like I decided not to do preterm pigs. I left that to Doug and Pear and Caitlin.
You want to say anything on that topic?
So putting my veterinary hat back on again, it's kind of difficult to model neurocognition, neurobehavior, because we will never ask any of these species to do math or taxes. I think the closest we can get and the best model for this is to go with like the more cognitively developed as they age. And that's probably, I mean, I'm putting on my Team Pig jersey again, and the stuff that Sharon's done and Ryan's done at Illinois is absolutely amazing. And I will never do that because the brain is a scary, scary organ. But I think that's probably, in a world of bad models, I think that's probably the best.
Paul or Claire, go for it.
Actually, it was a question from my fellow panelists. So is this model more geared toward assessing the positive effects of an ingredient on neurological development, or is it screening for toxicity?
It could do both. we've actually never gave like a toxic drug, but we've done, like I said, nutrient deficiency. And Ryan did a really nice study where he did prenatal and postnatal choline deficiency. So we could look at things particularly like during pregnancy or postpartum or both. Let me tell you, those pigs that were choline deficiency, they would not have found their car in a parking lot, you know. So and there were very specific prenatal things versus postnatal. You could rescue some with postnatal administration, but others not. So you can see deficits. Ours have mainly been through nutrient deficiencies. But yeah, you could do it with drugs.
Yeah, and I would say again, I'm not going to say we should be doing preterm pigs, but when you look at the neurodevelopmental clinical signs in a preterm pig compared to a term pig, they're very obvious. And so the ability to walk, their eyelids opening, just, I mean, and our colleague Per Sandgild and Thomas Tuman in Copenhagen, they've done a number of these studies and documented this. And so that's, again, it's back to Caitlin's comment about it's a great sensitive model to detect some of these things, but it's not necessarily context specific in that it's a preterm versus a term. So again, it just depends on whether you want to say, oh, we did this in term pigs, so it's applicable to term infants, which is, I think, is the major context of our discussion today. But the pig does actually can be sensitive for some of these neurological things.
I'm not as familiar with it or familiar with it. I obviously functional observational batteries are part of a lot of the tox studies we do, looking for toxic endpoints. I'm still not completely positive how relevant some of the endpoints in an FOB are to how does it translate to what does that mean for a human. Obviously, if your rat is staggering around and falling over, not a good sign. But we're getting into some very subtle, I think, changes, and how do you actually interpret that in a complex organism like a human? That's the catch.
So I'll come out in defense of Team Rat. So I did read through the OECD protocol for the extended 1-gen, and it specifically said, I've got it written down, the guideline places emphasis on neurological effects as the specific endpoint. So it is taking it into account. Now, is it the best model? I think that's where we could have additional discussion, and maybe to lead into tomorrow's discussion a little bit. One of the things that I don't love about the IOM guidance as it's written is it includes some endpoints for clinical studies that I think are not very practical. So like it is talking about putting infants into an MRI and measuring liver size. It's like, I don't think that's practical. I'm not sure IRBs would approve it. I'm not sure parents would sign up for it. But you've got something like neurobehavioral development. There's good clinical tools that are available to assess that you can do as part of a trip to a pediatrician's office. So like if you're talking about actually using human data for something, I mean, I think this is one element where you could have some discussion about, is this something that you tack on to a growth monitoring study and like actually just incorporate some of these cognitive tools, like, and that would give you the information that you need.
Anybody else? Any comments about that? Okay. Let's go to our second question here. Okay. Second question, and we touched on this. So should the field develop and publish a standardized model to outcome matching framework specifying for each major physiological domain that is model appropriate and which endpoint sets required and what standard should we create? And I think part of this gets to the reference that we touched on earlier about it. Is there a reference protocol, so to speak? But this question is, again, it gets back to model specific outcomes and how would we design those? So Laxmi, you want to jump in on that one?
Sure, I can try. And of course, I'll have help, right? So the basic outcomes that we always consider growth, as we all have been hearing about it, and then organ weight, length of the GI tract, and immune outcomes that we measure from all the different tissues that we can collect from these models. brain, of course, all the different behavioral outcomes one can consider. The question here is, do we have these reference data sets? I think Sharon answered with a big no earlier, right? And is it good to get these data out there published so we have a reference standard? Yes, absolutely. I think that would be required to have a baseline for some people to be able to go and refer to. It would be good to have that. Which endpoints, I think I've mentioned that, and which model is appropriate. I think I'm probably going to repeat myself. Initially, rat, I think, is ideal for in testing because piglet model is expensive, and neonatal piglet model is not I wouldn't say not fun, but at the same time, it's not just simply feed a pig like a rat would be. It is labor intense work. So I think you have to outweigh the benefits versus the cost benefit ratio there. So that's how I would go about standardizing the model and also standardizing the different outcomes that you're looking for. And it could be dependent on the ingredient that is being added. If it's HMOs, we know that it changes microbiome and it can have a gut-brain axis. And if it's a different component, what is that component specific effect in physiology? I think it has to be driven by that as well.
So can I ask Team Piglet?
Yeah.
Have you done any work on trying to develop an OECD protocol? I don't know what that entails, but I know it's a thing.
So that's a question. What would it look like, and what would it include? What would the battery look like? I think that's a great question.
So to do that, you'd have to get all the pig people to agree on something. And
What do you mean? We're all here.
there's tens of us, don't worry. And I mean, I think I also kind of break a little bit from Team Piglet in that, like, I don't think the pig is a good model for growth if you're testing a whole formula that's intended for infants, because piglets, especially the commercially bred ones that are meant for meat production, have been genetically selected to accrete protein to grow from a 2-pound bacon seed to a 300-pound monster in less than 6 months. So that's not going to reflect what an infant's going to do. I think the piglet is very appropriate for other safety, other function, other mechanistic questions, like that revolve around neuropathology, GI pathology, etc. So we already have a lack of consensus here. So I think, yes, there is an effort. We're working on it, but it's something that definitely needs to happen.
Yeah, I would disagree a little bit with that, because one of the values of the pig is that because they grow so fast over, just say a period of 2 weeks, they'll double their body weight easily. It gives you a better, more sensitive system, bioassay, if you will, to detect a difference, as opposed to an organism that grows slowly and grows at 15 grams per kilo per day as opposed to 60, which is about what a pig grows at, or more. Right. And another question, again, with regard to this point of reference, and we've discussed this among the pig people, is when we do a study in a pig, should we design those diets that we feed those pigs to meet their nutrient requirements, or should we take an infant formula that has the macronutrient content to meet the infant's requirement and just put that in a pig and see how a pig does?
The former.
The former and not the latter.
Correct.
Okay.
I agree with Team Rat.
Laxmi.
I wanted to add to your point, Doug. So we have done, it's about the whole formula itself. We have done study with, sponsored by a company, Elsa Nutrition, where piglets were fed with plant-based formula versus the cow's milk-based, dairy-based milk formula. IACUC would only approve where you have to add what was required for the growth of the piglet. So there's some limitations with that too, right? Like, can we add whatever that is there for the infant requirements, or are we going to have enough approvals for that? I don't know the answer, because
Well, we oftentimes we'll start with a sow milk replacer formula, basically pig formula. We can add some goodies that would be in a human infant formula, but not in a pig formula. So we do try to humanize it. But if you try to feed piglets human milk or human infant formula, you will have a malnutrition model. And if you're trying to look at brain development, immune development, anything, you're basically studying a malnutrition model. Our piglets double their birth weight in 7 days. But again, I would say little baby rodents grow just as fast. And basically, if you know little rodents, they're fetuses. Their eyes aren't open, whereas our pigs are up and running, and we can teach them to self-feed within a day. And so I think the rodent and also if you look at timing of brain development, with either the monkey, which is very postnatal, or the rat, it just does not align with the human, whereas the pig, the peak of brain development completely aligns with the human, even though the pace is faster, but when you scale it per body weight, it's much more similar.
Yeah. Well, what Sharon just said, we actually have done a study, and we fed them just infant formula, not at the pig's nutrient requirement, or we feed them donor milk, and they do not grow like a normal pig. I mean, it's perhaps obvious that would be the case. And so you can say, well, they are malnourished. So are you more likely to see or less likely to see a desirable or an adverse outcome under that condition? And I think you might argue that you could, because those animals are not growing and they're not quote unquote healthy. So there was a question in the back.
I was just wondering, sort of adverse events that we monitor in clinical trials. So how well does the piglet model work there? And so is there a pig alternative for every adverse event? Thinking in the digestive space.
Well, we can certainly see diarrhea. I've never actually seen a pig throw up. But we can see if there's gastrointestinal, we can see sickness behavior. We have scales to look at activity. We do daily temperatures. So, but yeah, again, a good thing to develop, standardized adverse event screening. If you're going to have a growth issue, like Doug said, you're going to pick it up really quickly because they're growing so quickly.
Yes, Barbara.
So the discussion around this piglet model, reminds me that we have several ways that standardized methods get established. There's AOAC, often regulations refer to AOAC. NIST gets involved with standard setting, and Cindy and I were talking, the NRC, I think it was the National Research Council, did actually recommendations for experimental animal models. And it seems, from what I take from what you're saying, there needs to be an approach here. It could make research less exciting because now you're following a standardized model, but to really have this piglet model be used in a regulatory way, it sounds like there's some need for standardization.
So there's a lot of need for standardization, but the problem with the pig model is that when you say pig, we also have different breeds of pig. We have our mini pigs, we're talking specifically about commercially bred animals, but there's also specifically bred mini pigs, the Ossiball models. There's a whole bunch of different subspecies breeds, whatever you want to call them. So that further complicates it because they have different dietary requirements that I'm not 100% sure are reflected in the NRC. So the mini pig diet that you get from whichever feed manufacturer you get, if you're looking at more adult animals, is going to hit the medium. It's going to hit like a potbelly or a mini pig. It's not going to meet the requirements of a commercially bred animal. So one of the challenges is what you're using that animal for. And there's not really a consensus on what in the pig and biomed space about which animals are most appropriate for what. We're in the team neonatal, so we're going to say the commercially bred pig is perfectly fine, but it gets complicated when you have other things confounding that. So yes, there are recommendations. I'm not exactly sure what they're for. I'm not sure if it hits the formula fed space that well, and that's where we're working on it, but it's not there yet.
Sarah.
It could be that they need to be updated or a new, to make it work, we may have to figure out where that pain point is.
We can lock some people in a room, but I've heard that's illegal.
Sarah.
Yeah, thanks so much, Sarah Maria. I was going to ask, I think we've had really good discussion about utility of the rat and piglet model potentially being complementary. There was some cautionary tales about, for instance, microbiome in the piglet and translation. But I was going to ask if we might spend a little bit of time thinking about NAMs and talking about NAMs and how they might work into this preclinical model for physiological outcomes. So enteroids and in silico modeling and what the thoughts are about looking towards the future.
So I'm going to take off my veterinary hat and put on my scientist hat because I do work a little bit with NAMs, and I think they can be very useful in specific contexts. It is important to note that everybody, I think, in this room is aware that in vitro and in silico models fail to recapitulate the complexity of a whole organism. So I'm going to take that and acknowledge that we know that, and say that there are a lot of things available that we can do a lot of high throughput modeling with organs on a chip that have been mostly used in the pharma space that have their champions and their detractors, but I think they're here to stay. And the funding mechanism, I live and die by the NIH because I was an idiot and went to academia. So there's more and more emphasis on including these in great applications. Great applications that only have animals in them are not getting funded, or they're not supposed to be funded. So one of the things that we're having to consider is like, where can we use these? If you're going backwards, if you're an animal person and you have to include these, but if you're more of a cell biology science person, like molecular biology person, these can be really, really valuable as an addendum to the animal work. So, and this is where I also put my academic hat on and say, you need a mechanism or a proposed mechanism, because otherwise you're just going, you can do as many omics on these cell models as you want, and you'll get a whole buttload of data, but it will not be useful to you. So it's you can treat them the same, not the same as an animal, but in the same context. You have to pick your model based off of what your question is, and that's important.
So I have good news that the standard genotox package that is run for all food ingredients, I consider NAMs. So it is cell-based, there's not an animal involved, it is the Ames assay and in vitro micronucleus, which is completely non-animal. So if your definition of NAMs is non-animal, then we're already using that for most food ingredients. But I'll endorse everything that Caitlin said. It's about identifying, are there endpoints that we need to look at, and then what is the best, most appropriate model to evaluate that endpoint. I'll also note, validation is also a critical element for these NAM studies. So just like we're talking about validation in the piglet study, it's going to be critical for these NAM studies, because I would hate for us to run a whole bunch of studies of NAMs and we don't know what the results mean, and then what do you do with that data? I think that's part of the morass that we end up with the current state, where sure, you can look through PubMed and find all the studies that have been done on all these ingredients, and all these in vitro studies will show you 1,000 different things. How many of them are relevant? Like, I don't know. Those aren't validated methods. I don't know how that translates into human. But if you do have a validated method like these genotox assays, then you know exactly what to do with the results when you get them.
Yeah, and I would just say on the NAMs front, the goal of the NAM is, new alternative models, is to get closer to reflect human as opposed to animal. And of course, in the context of enteroids, human intestinal cells, these are human. They're context specific in many cases. And we're just really starting to understand that. Like, you can take and make an enteroid from the jejunum or the ileum or the colon of an adult, and it will show you the differences in region-specific function depending on where that biopsy was taken. Or you can take a biopsy from a preterm infant, a term infant, an adult, and you can see differences that are developmentally specific. Now, in some cases, they may not necessarily be what you predict, and we're learning that right now, because we're literally, figuring it out. I mean, it's not like these NAMs or enteroids it's settled science, and because the enteroids, if you think about it, the cell models that we have, they're cell lines. CACO2 cell, HT29, whatever, they're cancer cell lines. Whereas these NAMs, okay, they're human also, but they are from different individuals. So you have to consider from a reference standpoint and a standardization standpoint, that makes it more complicated. Because you might say, well, do I have to do this assay now in an aneroid from 2 subjects, 3 subjects, 10 subjects? Does it have to be from a term infant? You know, one month old? So there are advantages, and technology is giving us the capability to do these things now, but we're still not there in terms of where we think we probably need to be in terms of standardization of the models. Yeah. Lakshmi.
I wanted to add one more thing to the NAMs. So there are these organ on chip systems where you can do like a single organ system or multi-organ. When I say multi-organ, you can have like a GI tract, liver, or like brain, where within the same system, when you add these bioactives or functional ingredient, the circulation of it, or immune cells, the circulation is to recapitulate what goes from the GI to the brain. So you could have like a multi-organ system, organ on chip system, so that will add some more additional strength to the mechanisms, and then address some of the things before you go to in vivo systems.
Okay, I think I'm going to try and keep us moving here and go to our quiz. Okay, for a triggered IOM tier 2 neurocognitive investigation, what is the most scientifically appropriate clinical model? We talked a little bit about neurocognitive, but this is just kind of an example of matching model to outcome.
Doug, I have a question.
Yes.
What would be an example of something that would trigger a tier 2 neurocognitive investigation?
Good question.
I was just thinking, like, I think Claire mentioned earlier, if you saw some sort of signal, that neurocognitive related signal in the safety study with a rat, you probably wouldn't then pursue it further because you would be worried about the safety of that ingredient. So I'm just trying to
Saw
understand.
a really profound change in your FOB, for example. You know, and that I used to work on pesticides. You would see that in a study of a pesticide. I have only seen that once in a study in 40 years on a food ingredient. So, you know, I think that it's rare that you would see that, but that's what I was saying. If you saw something profound enough that it popped out on an FOB, I would be hard pressed to think somebody would go, yeah, let's keep looking at that ingredient. That's got to be a good one, you know?
I think the other example would be if you have something in the literature that suggests from epidemiology that, so let's take DHA. So DHA is known to impact developing neurocognitive abilities. So if in a case like that where the literature is pointing to this organ system may have an effect, even if that is positive, you may want to take a look at it just to make sure that that positive effect still remains within what is considered normal human development.
Hi, this is Jen Simmons from Spherix. I often read FDA's questions at the end of a GRAS notice. So for a lot of the newer ones, you can see the FDA's questions for the notifier. And this whole discussion makes me think of one of the questions that they asked. I don't even remember who. I'm sure I've read it a couple times now. But the FDA is concerned about the long-term implications or effects of a particular ingredient, especially for infant formula. So let's say your tier 1 saw nothing, because Claire's right. Like if she and I are reviewing a rat study and the rats are twitching and can't get out of a maze, you're not going to pursue. But if there's something in the literature that the FDA is flagging in a question, or maybe during a pre-notification meeting, which we frequently do for infant formula ingredients, then it's good to have a plan in your back pocket, and you want to put your best foot forward. And so that's what I'm taking away from this discussion, is what is our best foot forward. If we get a question from a regulator, or if when I'm researching a new ingredient that we want to see, is this going to be appropriate, safe, and suitable for infant formula use, how would I test that if we have this kind of question?
And I was going to say, if you see something, you know, in an animal study that it's a flag, or even if it didn't show up, but your investigation into the functionality of the ingredient suggests that it has an impact on the neurological system. Those are hints that in your clinical study, you may want to be able to focus on endpoints that address that question, that this is not going to be adverse in a clinical setting.
Okay, I think let's move on to the next thread. Okay, the next thread, and I think for the sake of time, I'm going to skip the agreement and we're going to go right into the questions. So, when is the existing framework sufficient? So a clinical trial is required already in our framework of the IOM model. So the question is the mandated growth trial sufficient for physiologically active ingredients? And so our question is, question number 1, for safety, the existing pathway requires GRAS preclinical data plus a non-inferior growth trial. So for an ingredient with documented immunomodulatory activity in neonatal pigs, is the combination of a clean GRAS package, so preclinical assessment, and a clean growth trial sufficient evidence that ingredient's physiological activity is safe? Or is safety of physiological activity a distinct question that neither assessment was designed to answer? That's a complicated question, I'm sorry, but.
What do you mean by immunomodulatory? Because that could be good or that could be bad.
Right. It could be.
I mean, I think this is that example again of if you know about the ingredient, you know it has the potential to impact an organ system. Maybe it's a conversation bigger than what we would have in this room, but let's lay out what is the appropriate package to establish reasonable certainty of no harm. And in that case, would you want, I'll go back to the Callahan paper. So the Callahan paper recommended some immunological endpoints to add on to a clinical study. So I think I'm not going to make that decision on this spot right here, but I think in that case, it's certainly a situation where you'd want to consider adding additional endpoints to a clinical.
Yeah. I think to restate it maybe again, now that I'm thinking about it, it's like you do the preclinical and it's a functional ingredient, okay? Let's say it's a functional ingredient that has some well-known physiological function that you know of. And then you do the clinical trial and it basically supports normal growth. Is that all we need?
It depends on the ingredient. So I think if there is an indication that it's a case by case. So if you were talking about a non-absorbable, I don't know, DHA, I don't know. Does the DHA impact the immune system? All right, I'm not a nutrition person. I'll pick something else. Gauss and Foss. Oh my God. All right. Everything. Xanthan gum.
Let's pick one that we've been coming back to. Let's think about lactoferrin.
So lactoferrin. If it was lactoferrin, I think there's probably a good case that you would want to have those endpoints from a clinical. It integrates the Callahan endpoints into a clinical study.
So growth alone, anthropometry would be insufficient alone.
I won't say anything specific. I won't conclude here, but I think if you aren't going to have those clinical data, you'd have to have a rationale to support why you don't have them.
Yeah, I think the FDA would probably come back. And, as Barbara said several times today, you're not going to go through the extra expense of adding these ingredients unless you think there's some functional benefit. And it's not growth for lactoferrin. So if a company is thinking about adding that, they're going to look at immune function or disease, outcomes. So yeah, I think the Callahan paper does have some good guidelines. And if you've done the piglet study, then you've been able to assess all these other non-circulating immune pockets in the body.
Right.
So, yeah.
Yeah, I mean, we actually get into really dangerous territory here because there's a line between foods and drugs. And, despite the fact that, and I've looked extensively at lactoferrin, And I don't see any compelling evidence that it really has any, it does not have a pharmacological immunomodulatory activity. You can look at every study that's been done. Every time a phase 3 clinical study has been done, it failed. Why? Because there's no pharmacologic activity. It may have some kind of a more subtle immunomodulatory activity, and it certainly is an antimicrobial. But just through its physical activity in binding. But I think that we have to be very, very careful about defining what do we mean by immunomodulatory. What is it modulating? Is it a potentially positive effect? Did it create for an infant example? For example, for an infant, does it when it's in the infant formula, does it enhance the vaccine, the response to vaccines within the normal range? It's again, I think we have to be very, very careful that we're not actually looking at efficacy that is driving something that would be perceived as a drug.
Yeah, I'm parsing your second sentence, or is it this second clause? No, for an ingredient with documented immunomodulatory activity. So that's been documented. I'm assuming that means it's in the public domain, right? So the first clause of the second sentence, for an ingredient with documented immunomodulatory activity in a neonate pig. So that's documented. I read that to be published. And then there's a combined clean grass package. Well, that would have to be part of the grass package. Is it clean? How is it determined to be clean? If it's clean, why do we need to go on to the next step? So it's that. But the use is in infant formula.
Right.
So the question is very confusing, and it's somewhat contradictory as far as being a question.
I mean, just to add to what Dennis is saying, when you first put this out, the question I would ask is, what do you mean by a clean grass package? And did that clean grass package include things that address the physiological activity?
Yeah, so let me try and see if I can clarify the question. I'm sorry, it was complicated. But so let's say we have a compound that has a known immunological function, and we tested it in a pig, and we tested it in rodents, and it's absolutely safe based on all of our outcomes. But then we also show that it does have the effect of lactoferrin that we would predict it to have. Okay, it's not necessarily that it's a drug or that it's some therapy, but it's doing what it would be predicted to do. So then if you do a clinical infant study and you show that it's safe and supports normal growth, is that enough?
Hey, I was going to quick add, Ross from Helena, that I think this kind of gets to, so if we're looking at vaccine response or some immunological endpoint for everyone's favorite ingredient, lactoferrin, that's where we want to make sure we have normative values, right? Where, hey, if there's a little bump in the immune response to whatever vaccine they got, that's still within what we would expect for normal vaccine response to that particular vaccine. So that's where I think maybe a build or a takeaway from this is making sure we have those normative values. And I forget if the Callahan paper said those already exist or if we still need them, but that would be, I think, a great kind of objective way to make sure we're within a healthy functioning immune system.
So you mean normative values in the preclinical model?
It would be, no, we're talking clinical, I would say. Or preclinical would be great too if they exist, but I don't know if they do.
So I'm going to change the topic a little bit, but it's sort of related. So Paul, you've said many times, if an ingredient's not absorbed, then you don't have to do all these other measures. But I've done a ton of work in the piglet with human milk glycosaccharides and other prebiotics, and they are not absorbed. particularly prebiotics, but to a very, very limited degree. But a lot of their action is them being fermented by the microbiota, and then those products and other metabolites. So I think that a lot of discussions around how safety is assessed we're not looking at it through more of a modern microbiome lens. And I understand it's like stepping into, unknown territory because we don't even know what a healthy microbiome is. But that doesn't mean we ignore it, right? Because if you're saying we want to add this because it's bifidogenic, and we know it's bifidogenic because these bacteria utilize it as a fuel source, and they're making metabolites like acetate, lactate that can have beneficial effects. I don't think we're not adding them as a, neutral, or what's the term, an ingredient that's not having a systemic effect. So to me, we have to, in the terms of infant formula, because those are the outcomes, we have to change that lens a little bit.
Yeah, and I don't want to turn this into the microbiome conference, although I think we could probably do that pretty easily. So the comment has already been made. It's already really hard to define what a healthy microbiome is. So when I think about the microbiome, and actually I think the FAO had a document that came out not so long ago that talked about using the microbiome as an endpoint for safety determinations. And they more or less conclude that it's too early to use it right now as an endpoint. And to me, I totally agree with everything you said, Sharon. Like, you're right. Like, the microbiome is doing things, and it could have effects that are not within the GI tract. So I think, to me, that may point not towards we have to do more on the microbiome, but maybe it's an indication that we always want to run an OECD 408 or an OECD 422. So those studies will pick up effects even if they're not in the GI. So if they are creating metabolites that then go to the liver or the kidney or et cetera, that would get picked up in your general tox study. Changes to the microbiome, we do not know enough today to say what a change in the microbiome would be considered adverse.
Yeah, I don't know whether we can say that this is what a normal infant microbiome should look like. Would you?
We know what abnormal looks like, I think, in some degrees. But my concern is if you're giving HMOs to an adult rodent, there's very few bugs there that are going to be utilizing those. There's some, but so it gets into that. And I think we've had comments about it, even as the pig microbiome, the same. But you can humanize. We've done this with piglets. You don't need germ-free facilities. You can put human infant microbiome. As long as you feed them HMOs, you're going to get that bifido to grow. Sometimes that's an experimental, but you have to have the right microbiome with the right fuel source. And if a company wants to add this ingredient for that outcome, then I think it just has to be incorporated into the gut-brain axis and all of this. And this is why some drug trials have failed, or you have adverse because they haven't necessarily included the microbiome. And we know that, for example, things like metformin and other drugs is significant microbiome modulation of physiological outcomes. So it's not just infants. I think the whole FDA safety of drugs and foods we need to figure it out, right? We can't just say we don't know, but who's going to pay for it? Yeah.
Question.
Yes, Sebastian Finch from Art of Foods and Ingredients. I just have a general comment for people to reflect on, especially after that last comment from you, Sharon. I would like us to try and think about tangible progress from a manufacturer of ingredients. Undoubtedly, it's like trying to peel an onion here. So we have scientific progress going on, and you think about adding constant extra layers, which is fantastic, right? We're progressing. We think of new safety concerns, and we want to address those. But at the same time, there has to be a balance where the constant scientific progress doesn't stop the evolution and progress that we can make in the short term for infant formula. So how do we also balance that? Yes, we should constantly pursue the fantastic goals we have long term, but by the time we get there, there'll be new goals, and we shouldn't pause and wait to keep establishing at the next layer and the next layer. So a little reflection to to make constant progress and not to try and reach 100% or as near as we can to that full safety. And by the time we get there, there's new concerns and new ideas we have to address.
Don't let the perfect be the enemy of the good, right? For progress. Yes. Okay, I'm going to move on to the next question. Oh, Sarah, go ahead.
Yeah, thanks so much. We have a comment from Robert DiGregorio, and I think it actually speaks to what you're a little bit to what you're talking about. So he's speaking, I think, about thinking about our definition of safety and also thinking about post-marketing surveillance. So he's basically commenting that we need to reconsider how we use the term safety. Safety isn't proven even in a clinical trial, but absence of safety signal can be demonstrated. So I think he's making the comment that post-marketing surveillance is would be a enabler of actually moving product into market.
Great, good point. Thank you, Rob. Okay, our next question for this thread for suitability. Okay, so for a growth trial demonstrating comparable growth and tolerance, it does not demonstrate that a functional ingredient's intended activity is occurring or is beneficial. Okay, for an ingredient with an intended microbiome or immune development claim, what clinical evidence is needed and is it compatible with growth trial design? Now this gets a little bit into tomorrow's, but I thought let's just throw this one up here for now.
All right, so let me start this one. So I'm going to start with, it's really important as we have these discussions that we have to keep these two elements really separate. So if we're talking about the evaluation of safety, these are questions that are not in scope for that evaluation of reasonable certainty of no harm under conditions of use. And I bring it up here and I'm trying to make it emphasize this point a little bit more because I think we do start seeing these two circles of the Venn diagram overlapping sometimes when we have these discussions is, oh, you're talking about claims, oh, you're talking about benefit, it's a drug. Whether or not something is a drug is also not part of a determination of safety. But is something a drug is a definition that is completely separate from is something safe or not. If we're going to define drug as anything with physiological effect, again, let's throw in essential nutrients and technological additives and all these functional ingredients. That is not a helpful definition of drug or not drug. Tony's not here, so I'm going to speak on his behalf, and he can correct me later, or maybe Dennis can. But the law defines what a drug is, so I think we have to be really careful when we're having this discussion and the one tomorrow. Like, we're not having a discussion of is something a drug or not when we're talking about safety. Talking about safety is the standard is reasonable certainty of no harm under conditions of use. Do you want to add acetaminophen to infant formula? No, you can't. It's a drug. Like, we can look to the law and we can look at that. But all right, sorry, brand complete.
Sorry, follow-up question. How is a drug defined in the law?
It's a substance that's a subject of substantial clinical investigation before it has been used in food. Oh, yeah. Thanks.
Yeah. Magic 5 words.
Yeah.
I was just going to look it up.
Yeah.
I was just going to look it up, but it has a legal statutory definition, and when regulators are deciding which box you fall under, they're looking at that drug definition, they're looking at the food additive definition, or they're looking at the dietary supplement definition. Intended use, right? So the same chemical could be, depending on its intended use, could be a drug. It could be a food additive.
Yeah.
It's not an inherent property of the substance.
No, there are some things that are actually both GRAS and were investigated as drugs, as long as you do the food before you do the drug. Yeah.
Think caffeine.
Oh, yeah.
Yeah.
So
I have a question if we're done with the drug. Sorry, this is Jody Butler from Nestle Nutrition. I think the Callahan paper has come up, and maybe park me if it's going to happen tomorrow, but to me, it's a long list of criteria. So it is about safety. It's not about efficacy, but when I look at it, I don't see the must-haves. So I don't know if we're sticking with safety from that list that, the paper's been published a couple of years. Has it been narrowed down to like you have to look at IgG, IgE, or is, I see a list of things and directionally we know either they go up during infancy or they kind of stay stable. We may know whether they're similar or not similar to breastfed infants, but from that list, is there any progress on shortening it to what we think? If growth is not adequate, then you at a minimum have to include something like, I'm making it up, IgG, IgE. I don't know if anyone knows if there's been progress made, if we specifically are talking about safety, not efficacy.
I don't think so.
Because it's hard to refer to it because it's a large document, right? Like if we say the Callahan paper, it's quite the endpoint. The clinical endpoints are long and the laboratory endpoints are. It's a long list.
Well, we'll talk a little bit about that tomorrow in session 3. I mean, session 3 is all about clinical endpoints and basically this question enhanced for 2.5 hours. But I will say we did ask that specific question to our panelists in that pre-workshop survey just to see where consensus stands. And there did seem to be some alignment on at least ranking, prioritizing the bulk of these endpoints that were identified in Callahan. And so we can talk about that a little bit, but this is an N of 1, essentially, the INSC. It would be really great to talk about, again, a roadmap for how to develop that consensus.
So I'm hearing then the answer to question 2 would be that it depends again. Don't do it. Yeah. Okay.
But I would like to ask for both of these. I mean, the question for session 2 panelists is, when would preclinical not be enough where you would need to go into a clinical trial, either for safety or for suitability? For any of these, physiological outcomes we have been discussing today.
Well, you're talking about suitability, or are you talking about supporting marketing claims?
I think in this definition, we are talking about marketing claims. I mean,
yeah, I mean, the second part of that question is not safety, and that's not one that we would deal with. But if you're talking about what do you need to do to support a marketing claim, FTC and FDA have a lot of guidance on the level of clinical data that you're going to need to do that.
Then you can go back to that first question on safety. We can just focus in on that one. Still, the question remains, when do you feel that some of these questions cannot be fully answered just on safety alone? Like we know already the standard clinical or preclinical package should be sufficient for GRAS determination, but we also know that it does not, just based on precedent. So when would you see the need to move from preclinical studies to a clinical for safety determination of an individual ingredient, not infant formula? And the basis for this question is that clinical trials are already required, but that's only for a finished infant formula. It's not required for an ingredient. And yet it seems like there is an expectation that some clinical evidence is needed to support safety conclusions of an individual ingredient.
Well, I think that depends on the results of your preclinical. If you found nothing in a robust preclinical package, there's no question you're going to ask in the clinical from a safety perspective for the ingredient. The clinical is then testing its safety or nutritional adequacy in the finished formula, which is a different question. So, the need for a clinical would be driven by, in the research we've done in the preclinical and the literature, it suggests that there is an endpoint that is going to be too subtle to be picked up in a preclinical study. So we think we're going to have to actually submit clinical data to show that this is not going to be a safety concern. Example would be human lactoferrin, right, where they asked a question to which there is no preclinical model because you cannot study autoimmunity in an animal with a human protein. It just that doesn't happen, right? So, but I don't think there's all that many situations where a clinical study turns out to be mandatory to establish the safety of the ingredient.
But I think, aren't we talking about here in terms of the necessitating clinical trial? We're talking about clinical trial in infants, right? So if you said, okay, I want to test an ingredient that is destined to go into an infant formula, and it's got to be in an infant clinical trial, you're going to put it in formula.
It's going to be a finished product.
Yeah,
Right.
it has to be.
Can't
Yeah.
feed it any other way.
It
Exactly.
has to be. Yeah.
Yes, but it needs to first go through the GRAS notification process.
Right.
It needs to be
Right.
reviewed. So that's
Yeah.
where I'm focusing this.
Yeah.
Like, yes, of course, we know that
Right.
in order for it to get to market, it will need to go
Mhm.
into a clinical
Right.
trial, but it can't even go into one
Yeah.
unless it is first. It can't,
No,
fix my language, Paul. It can go into a trial, but the necessitation for it to be included in infant formula is that it must go through the GRAS review process. It must
Right.
have undergone review and been determined to be safe by the manufacturer with no questions from FDA. But that's where it seems that there's ingredients getting stuck, not on the, they're already approved. I know that's not the right word, but they're in a clinical trial and they're having some problems there. It's on the ingredient side, not the finished formulation side.
You're asking a sequencing question. So like we all know you're going to have to do a clinical at some point. So do you need to run the clinical to support the GRAS evaluation, or can it wait to be part of the growth monitoring study that's part of the formula notification? That's the question. So are there cases where there's enough outstanding questions around a specific physiological effect that is not adequately covered by existing preclinical models that we would need something additional?
Yeah, and this matters a lot on the practical level. I mean, if we're going to industry, there's not one industry that's doing this, is the suppliers and the finished good. So who's taking that responsibility for an individual ingredient evaluation if it's determined that a clinical endpoint is needed? Does that mean every ingredient supplier now needs to partner with an infant formula company? And how translatable are those results if it is within the matrix of an infant formula to the applicability of that individual ingredient in other infant formulas, right? There's a lot of complexity, I feel like, with this question. It's not just sequencing, but also who's responsible and what's the appropriate, yeah.
Yeah, I mean, I think that, depending upon what else is known about the ingredient, that really ties into what FDA is going to look for, or what an expert panel would look for, what scientists would look for to determine that it's safe. And I promised Jen I would use this phrase. We call it the principle of convergent validity, made up by my friend Peter Pressman, which means that you're not necessarily taking data from one particular thing. So you might look at animal studies, you might look at what's known in vitro, you might look at someone has done some models that spoke to the physiologic activity. You're going to look at the metabolism and the fate and the activity of that ingredient. And is there a plausible mechanism by which you think there's going to be an adverse impact that cannot be ferreted out and supported by all of the research that you have looked at? Do you need that clinical study? Something that is completely de novo, that has not been in the food supply, that nobody knows anything about. I'm thinking FDA is going to feel a lot more comfortable if you had a clinical, right? Because they're going to go, geez, nobody's ever been exposed. If you have something that has been in the food supply for ages, maybe only in adults, we have a lot more that we know about it, and that we can look at and determine what would be a plausible mechanism by which it might produce an effect or not. So again, I think case by case is the answer to every question we've had today, but I think that's what drives it. And, and I've worked with clients who do their clinicals before we get to the GRAS, right? It's a safety evaluation with an IRB, and they do the clinical before they do the GRAS, and many of them don't do a clinical, get the GRAS, and then go to market, and then, whoever is putting the formula together does the clinical for the infant formula notification. So it can be either one.
Yes.
I just want to re-emphasize with something I said earlier. It's generally recognized as safe. And so we used to joke that if you have to ask, it's not GRAS. So it needs to be obvious, and the science needs to be well established and not experimental. The assays need to be well accepted and validated. So in a situation where you have a truly novel ingredient, the GRAS pathway really probably is not your pathway from a regulatory perspective. It's a food additive. And there are food additive regulations for use in infant formula. So everything doesn't go through the GRAS process for food ingredients used in infant formula.
And the question is, how do you define something that's truly novel, right? Like if you're talking about a recombinant protein that's similar in structure to a human milk protein, is that truly novel or not?
Well, and those sorts of questions are resolved with a pre-submission consultation in terms of GRAS. And there's a discussion about, is that information generally available? And is there generally available information to justify that they're substantially equivalent?
Barbara, you had a comment. Oh, Bert, I'm sorry.
I'm getting a bit confused by the discussion, I admit. If I look at this scenario here, the scenario is the decision was already made for doing a clinical trial. And you have a situation where you are pretty confident from your GRAS data, from your neonatal piglet data, that the component is likely to be suitable for use. And then you do a clinical trial. Now, with a clinical trial, you have a data monitoring and safety board. You will detect, if you do it properly, adverse effects during the clinical trial. So why do you do a clinical trial prior to the clinical trial? I don't understand this really.
Like you'd really only do one. So it really comes down to like, it's a sequencing question. So like, do you need to do it to demonstrate safety of the ingredient?
But the scenario says the decision has already been taken. A clinical trial is required. That's the scenario, right? So then we have all the prerequisites. If you have the data, the clinical trial board, the IRB will look at the data and say, is this reasonable to be tested in a clinical trial? And then you go ahead.
Well, the clinical trial is required for the infant formula notification. It's not always required for the GRAS status.
Sure, but
Right.
I'm just referring to the scenario that is on the screen.
Right.
I think there's several things that are getting wrapped up in this. One of which I will comment that I think one thing that made it hard for FDA was the practice of embedding a GRAS notification into the infant formula notification. They have different timelines. They serve different purposes. And it often put FDA in the position of having to decide on the notification before the GRAS notification had been completed. So it didn't leave everyone feeling good about each other. Let me just put it that way. I think the other thing that's getting confused is there's that requirement for a growth monitoring study, which I find is just easier to refer to it as the growth monitoring study than to refer to it as the clinical study. Because once again, why are you putting this in? You're putting it in so you can make a claim. And there will have to be evidence to support that claim, which is likely to be a clinical study. So again, it just feels like there's several things that are getting kind of tied up together.
Yeah. I'm going to go to our quiz, and I'm going to throw out a little question here while everyone is answering our question here. So for a novel ingredient with documented activity in a neonatal model, is GRAS preclinical data and a clean growth trial sufficient clinical evidence that the ingredient's physiological activity is safe? So I would ask a question on top of that. And so that was one of the first points that Paul touched on. So if you have a new ingredient that you consider a functional ingredient, you've done the preclinical testing in 2 models, you've shown that it's safe, actually has some physiological function in those models, say it's a pig, and then you do a growth trial and you don't measure anything other than growth, does that preclude the ability to make a claim about that ingredient?
Oh, now you're mixing up claims again with the other things.
Okay. Yeah, this is for my clarification. Yeah, you're saying the answer is no.
Yeah, we're not talking about claims. And I'm not saying it isn't an important discussion, but when we're talking about safety or even suitability, I think that is a different discussion than talking about claims. And Barbara's right. Like you would need a clinical study to support any claims that you're going to make. But I agree. I think it's helpful to think about that separate than there is a regulatory requirement to run a growth monitoring study. Here's the endpoints that you have to conduct. Like this is what you have to do. That, infants grew fine is not going to be your claim. Like,
Right.
well, I mean, it could be, but.
So what you're saying then is if you wanted to make a claim, you have to have an additional trial where you have appropriate endpoints to show that the claim is substantive.
But I thought you said that you could add immune outcomes, like from obviously the Callahan study. So you could meet the needs of the growth monitoring study and have some additional outcomes that could get to immune or microbiome or something else.
Yeah, correct. And even if you ran a trial to look at potential benefit, I mean, you're going to include some of those anthropomorphic measures just to make sure nothing's going wrong anyway.
Yes, Bert.
Bernd Stahl, Danone Research. We had this discussion in Europe. If you would have a growth study and you have extra parameters, usually these are secondary parameters, so they are not sufficient to get a claim. So you can get insights which could help you for another efficacy trial afterwards, which is designed and powered for the health outcomes. So I think it would be, it's useful to have that in a growth trial, but maybe not sufficient.
Okay.
Yeah, I think that's a good point, because when you do a trial, you have to power it for the outcome, and you're probably not powered for your outcome in a growth trial. Your goal is to actually get the growth trial done and get the infant formula notification, through.
It could be powered for some of those secondary endpoints, like microbiome, some of the gut biomarkers. You have enough power in the numbers you required for growth and safety to see some of those endpoints. Not probably like reduction in illness risk or medication use, or maybe even that's more iffy. But some of the biomarkers, you're powered if you go with the typical growth and safety.
Okay. All right. Let's move on to our last thread here, and our last thread is about ingredient interactions and emerging technologies. So when multiple bioactive ingredients co-occur in formula and a new production technology advances, how should the preclinical framework adapt? So for a reformulation and adding a novel functional ingredient to an existing product, is the scientific unit of evaluation of the novel ingredient alone or in combination with existing bioactives sufficient for the finished reformulation? Should the answer be different for safety or suitability? Sure. She's answering. She's got an answer.
Well, so when someone comes to us and wants to do a piglet study on human milk oligosaccharide, I try to convince them that if they have other functional ingredients in their formula that they do not intend to remove, in their final product, that we should include those. So if they have nucleotides or even things like taurine, which isn't going to be in a pig formula, some will do it and some don't. Some want a very clean study. I just want my ingredient. I'm just going to do that. The nice thing, again, in the piglet, you can do 2x2 designs pretty easily. So like if it's a prebiotic, probiotic, symbiotic study, we can look at each one individually in combination and different doses. But yeah, I would say that I typically say have what you think you want in the final ingredient in your preclinical study, but not all sponsors have agreed with that. They want a clean study, I guess.
Yeah, I would submit that this is not even capable. We're not even scientifically able to do this in regular food. To look at interactive effects and with all the different components in food, and to make determinations about safety. It's always based on individual ingredients and the safety assessment of the individual ingredient.
I wonder if anyone could speak to the DHA ARA example, where those are two separate components but needing to be investigated as a complementary pair.
I think, I'm trying to remember way back then, but I think that they had to be in combination because ARA alone produced a detrimental effect. And so that resulted in having to test them in combination. You know, it's an interesting question about the combination because we've asked FDA many times, and we've actually had discussions with them about testing singly versus in combination with something that's already the sort of safety been supported, and we want to add another novel ingredient to it. There is, I'm not talking about in the food itself, but I'm talking about just testing in a rodent bioassay or piglet bioassay, the two in combination, and there's a benefit to doing that in that if there's going to be any kind of a synergistic or antagonistic or potentiation effect, you would see it in the ratio in which you're going to give it. So especially if you're going to give two things that act via, they have the same functional capacity, and you want to know whether or not you are still within a normal physiologic range when you add them in combination at the dose you want to add. So, I think typically if you can do them together, but you would always put, I would put within that bioassay, I would put an arm with the new one alone, and then have the combination, and then you have the control. So you have to have the right controls so that you can tease out the effect of the new one from the combination.
Right. So maybe it wouldn't be required to study that ingredient in a phase in phase 1 or tier 1 type of studies with the other bioactives, but maybe it would be helpful, optional. Yeah. Any other comments about that?
I even wonder if you could propose not a tiered, a decision tree approach, much like we've been talking about with physiological outcomes, where tier 1 leads to tier 2, if only there's a trigger. Same thing here. If an ingredient, in the IOM guidelines, first you do a literature search to say, are there known or putative physiological effects? And that would lead to some investigations in your toxicology model. Going back to this question of synergies or antagonistic effects, could we propose maybe we say, if you are investigating a single ingredient, first do the literature, find if there are any known or putative interactions, and that would then lead to a decision tree matrix, right? So it's not, everything we've been talking about today is, it depends and it's case by case, but can we reframe all of these conversations into, we should establish a decision tree process so that it's a more systematic approach to determining, yes, we should do this, or no, we don't need to.
Well, and I can think of an example where 2 novel ingredients, you would want to maybe put them together. So think of a prebiotic and a probiotic. So if you want to look at the safety of a probiotic alone without any, say, HMOs, say you're looking at bifido or something like that, you might want to do that alone. And then if you're proposing to add an HMO, you want to look at it combined, because you might anticipate that there's going to be some synergistic beneficial effect of having those 2 novel ingredients together. So I think there are circumstances where it might be very useful to have combinations. Yeah.
But I think you're right. Like, so if we're mapping out the roadmap of what do we want to put into this guidance on how to develop your safety program for your novel ingredient, making sure you're considering and identifying those situations where you may have interactions with other known ingredients, I think that's something that we'd want to build in. It wouldn't be everything, but there are certainly cases where there are known interactions.
Right. Okay, let's go to our last question here. Cypress says I've got 5 minutes. So. No, let's go to the second question, just to Q2, not the quiz yet. Go back. There you go. Okay, what validation evidence would be needed for a neonatal intestinal organoid assay could contribute meaningfully to a safety or suitability determination? And could a validated organoid platform eventually substitute for any component of the tier 1 or tier 2? Caitlin, you want to jump on that one?
So we don't understand a lot of how organoids and interoids work. For example, in my own work, we've been working with preterm and infant lines of interoids, which are basically small mini guts in a dish. And we just now discovered how to inflame them so we can test anti-inflammatories. And we've been working with these for years now. So I have a hard time even seeing how we would validate something to be able to include it in this regulatory landscape. But weirder things have happened, and that has, the organs on a chip have been integrated into certain pharmaceutical validations and testing, and I think it's very possible. I think we need a lot more data about how these behave and what kind of information you're looking for them. And that, as I'm going to say, as an academic, comes down to the mechanism of action. What is your ingredient trying to do? And then it might be useful to put it on an organoid or an interoid assay.
I think if you look at the success stories of where NAMs have been validated for certain endpoints, they're very narrow. So you look at skin sensitization, I think is the most recent one that just got OECD validation. Something like that, something about the BCOP, which is like a bovine cornea irritation model. There's a lot of very specific models that have been validated, but like they're very specific. So it's back to, we would want to be identifying endpoints that we don't have really good models for. And then we would absolutely want to consider, is there a NAMS approach that would be the best way to address this question?
Yeah, I agree. I think it's still too early to tell whether these organoid models are going to be better than the existing models that we have, that are cell lines in most cases. So it would be very difficult, number one, to take time to establish these references, even if we do. My microphone just died. So there we go. Thank you. Yeah, we're running out of gas here. So yeah, I think too soon to tell. Thank you, Cypress. So any other questions maybe about the organoids or the interoids from the group or online? Laxmi, do you have any comments to add on the NAMS front and organoids and interoids?
No, I think it's covered pretty well. We don't know, especially when you combine multiple bioactives or multiple functional ingredients into the NAMs. We don't know enough yet.
Yeah.
Hopefully we will soon.
Right. Okay, so with that, we can go to our last quiz here. There we go. For reformulating and adding novel ingredients or multi-ingredients, what's the appropriate scientific, I can't read that, unit? There we go. Yeah. What's the most appropriate scientific unit of evaluation for examining multiple ingredients in the context of safety or suitability? It depends. We'll put that as the last box in the algorithm of the decision tree.
Depends on who's reading it.
Okay, well, so now I get to try and summarize what we've just talked about for the last 2 hours, and I have not been taking notes, so I'm going to do my best. I've been taking notes in my brain. So I think one of the first questions that we had was with regard to safety, and are the existing models that we have, the rodent models that are set forth in the IOM, are they sufficient as a starting place for evaluating safety for ingredients for infant formulas. And I think we have kind of a lot of agreement on that, that the existing framework that we have in place works. But then when we consider tier 2 evaluations of ingredients that might have a physiological function, that the pig is a useful model, a very useful model, an appropriate model to evaluate many of these, but probably does not get to the point of where it's a mandatory model that we would recommend, that it is mandatory for evaluation at a preclinical stage. And then in terms of, you know, would using the preclinical models, including the pig, and showing that it has a physiological effect in a tier 2 evaluation, and then showing whether or not that ingredient is safe in a clinical study, is that sufficient to basically be able to identify that formula with that new ingredient as safe? And I think at that point, we feel like maybe that clinical study needs additional endpoints included in the evaluation. And then finally, this last point here, do we need to evaluate novel ingredients together with other bioactives? Not necessarily. When it's appropriate, probably we should evaluate those in separate arms of preclinical studies to evaluate their safety. And then finally, on the topic of NAMs, I think it sounds exciting. These are new models that we're learning about, but it's still too early to tell at this point whether or not these are going to be sufficiently validated and standardized to be able to replace some of the other preclinical NAMs models that we have. And then one point, I guess, that coming back to the pig model, I think there's kind of general agreement here that if we are going to include the pig model in the tier 2 evaluation of ingredients, that we really need to identify references and create a reference framework for doing these studies so that they can be comparable across different labs and different conditions, so that they can be useful as references. And there's various regulatory frameworks to follow that have been recommended to use as a guide for that. So that concludes. Does the panel want to maybe add anything that I left out? So I want to thank the panel for a lot of hard work answering those questions, and really had a great discussion today. So let's give everybody a great round of applause. Laxmi, we appreciate your participation today. And at that point, Cypress, I think that closes our activities for the afternoon. Thanks, everybody. We are adjourned.